All right, good morning, everyone. Thanks for coming to campus on a Saturday. My name is Matt Sikora. I'm an associate professor in pathology. I run a research lab on campus dedicated to invasive lobular carcinoma, and we are really excited to have you all here today for our Front Range ILC Bootcamp. This was designed kind of in preparation for the big ILC Symposium next weekend, but whether you're attending or not, I think what we're going to cover today is going to be a lot of great information on not only how to on one end, like empathize with the scientists. We're as scared of you as you are of us. And then we're going to talk a lot about what you might hear about at a breast cancer conference. I'll focus a lot on lobular biology, and we'll try to decode some of the jargon that undoubtedly you've heard about in the context of your diagnosis. And I want to acknowledge that all of these events we've been doing so far on campus, this is our third, which is fabulous. It's kind of cool to say that. This has all been supported so far by a grant from the Breast Cancer Care and Research Fund, actually out of California, and the current president, Laurie. We're also really grateful for the support from the Cancer Center. Thank you to our admins showing up on a Saturday and volunteering to help out. My lab, I have a couple of folks here today. Project Bridge we'll talk about, and then also this is supported by the CU Entrance Lobular Breast Cancer Research Fund, which was established by the late Lee Pate, Laurie, and several of you in the room have supported the fund. All right, I'm going to give a quick overview of what we're going to talk about today and then hand off to Front Range ILC for a little bit. So we'll kick off with Donna and Tracy talking a little bit about what we're thinking about, where they want to go. I just try to facilitate. This is all about what your group wants to do, really. I'll talk briefly about, we're having this conference next week. Why do we have scientific conferences? Kind of where do they sit in the research life cycle? What's the point? They're not just for vacations. We actually know it is business. I'm really excited today to hand off after that to Project Bridge, which is one of our, I would say, the premier student group on campus for the graduate and professional students, with Devi and Laurie Lyon. They're going to talk about how to attend a conference, what to think about in that space, and how communication is set up very differently for scientists and for literally everyone else in the world, and how to navigate that. I will then yammer for a while about cancer biology, breast cancer, ILC. I'm not going to talk a lot about research today, but I'm happy to anytime. But I'll focus as much as I can on basics, terminology, and again, try to get everybody up to speed for what you're going to hear about kind of at a base level at a research conference. And then I'll go through a little bit about the conference proceedings for next week. Even if you're not attending virtually or in person, it is all going up online after the fact. So I'll try to cover some highlights, why some of the sessions are there, etc. And then I'll hand back off to you all about what Front Range ILC wants to do moving forward. So with that, we'll start.
So hi everybody, I'm Donna. And we're nominally trying to pull together this Front Range ILC. What it is kind of part of the discussion. I've been working about a year or so, I think, on kind of getting this going. And the main presence that we have is the Facebook page. So if are you all connected to the Facebook page? If you're not, we can help you get connected. It's a great resource for information about both lobular breast cancer and just support as well. Because it's all very challenging to navigate through this. Do you want to say a few words?
There's the page.
Sorry, yes. If you're not part of it, we will help you become a part of it. Come and ask questions about the diagnosis, your providers, where you got care, things you need for pre and post-op treatment, all that stuff. We want to bring sort of the national education on ILC to our local folks. You keep folks apprised of what's going on here in that lab, especially because it's super exciting to have a dedicated lobular research lab that is not a normal thing in most of the country, and we're very lucky to have that here. And so part of this group forming is to just kind of make local patients aware of what a unique resource that is, so that we can all help each other out. Hopefully soon we'll have some really robust ILC clinical research trials going on at CU Entrance, which is my dream. And then also just a quick plug also for the Lobular Breast Cancer Alliance. I'm the chair of their board. They have a lot of resources. We share those resources with the local group. And if you need help sort of outside the Front Range, they're a great go-to organization to get started. So thank you for being here. Yeah, so things to think about while we talk today, we can circle back on this at the end, but like going forward, what do you need from this group? What would you like to see it do? How can we serve you the best? is getting together in person, doing webinars, what is the best way to kind of reach everybody, maybe even in the Colorado area? And then are there certain activities we want to focus on, given that Breast Cancer Awareness Month is around the corner, and there are a lot of feelings attached to that month, but certainly it can be a good opportunity to make people aware that breast cancer is not only one thing, that there are subtypes, and Lobular is a big chunk of the patient population. So just think about this as we go through today, and then we'll get you feedback.
And I'll put a plug in for, I like to actually say that the A for breast cancer, one should actually be action on, because we need to do more and be much more proactive than just being aware. So I think there's a lot of opportunity with that as well, especially with social media, and so maybe we can talk a little bit about that. And
We're all very aware of it.
yeah, we hear that. And when I moved here 14 years ago, when I went to register my car, they're like, would you like a specialty license plate? And this is right after I was first diagnosed. And they were like, oh, we have one for breast cancer. I'm like, oh, fantastic. Where does the money go? And they were like, oh, no, thank you. So I think those are the, we're all aware, we see everything, but we need to move the needle. But so maybe think about these things, like Tracy said, and then we can talk a little bit more later.
Like we're talking about the inspiration for this particular Front Range ILC meeting was the conference next week. So let's talk about the relevance of that. Before I get into that, though, I will introduce the rest of the lab. So you met Sydney and Maddie. Some of you met at the last session. So lab manager and instructor in the lab, Joe Sotnick, did his training at Colorado State. He does all the stuff in the lab related to the estrogen receptor and its function in the genome. Jordan Swartz is a post-bac scholar. She's also, she did her training at downtown. She studies more DNA damage response in lobular breast cancer and some stuff where we're going in new therapeutics. Sydney is not only going to do some stuff with advanced models of ILC, how we can better mimic what ILC does in a patient, how we can do that in a lab better. And it also, both Maddie and Sydney are studying places where what we're learning about lobular cancer actually seems to overlap with ovarian cancer. And since lobular can metastasize there, we think that's no coincidence. So we have some new projects studying the fuels that both of these cancers use, what they eat, and how that helps them grow. And that could be another hour if I just keep talking. Maggie Music is a postdoc in the lab. She did her training. You got her PhD at Clemson. She's studying where most of the lab has been studying kind of what cancer does when it already exists. She's taking a step at it and asking, how does lobular cancer happen? What are the genetics of how lobular cancer starts from a normal breast cell? I'll talk a little bit about that. Orly's not with the lab now, but she was a summer undergraduate fellow. Oh, you can't see my pointer. There it is. Orly was a summer undergraduate fellow from Brown that did some work with Maggie. And I covered Maddie, and there's me. I don't actually do any work anymore. I just write grants and talk to people. But quick nod to our funding. We have funding from the National Cancer Institute, the Department of Defense Breast Cancer Program. Dynami Foundation is an outstanding ILC-focused foundation that's based out of Michigan. Cancer League of Colorado, if you've seen their events, they're an outstanding funding organization for cutting-edge research. Can't say enough about that group. We have funding from the American Cancer Society and then the Lobular Breast Cancer Research Fund. Since some of you heard about our research at the last, just quick updates. Since April, Maggie in particular has been pretty busy. We're talking about conferences, so she had the opportunity to present her talk at one of the premier meetings on hormone-dependent cancers. We think about estrogen and progesterone. She represented the lab on her project at what's called a Gordon Research Conference, which is a very small, but basically all the thought leaders in this field, and she got to present our work, and she represented us there. She also received a second year of support from our campus's cancer biology training grant, which is a really awesome program that gets postdocs and graduate students funding. So then I don't have to worry about it. So I can spend the money on other things like research and hiring more people. So it's great for her. It's great for me, great for everybody. And then again, I'm not going to go in depth in our research today, but we are going to have 4 posters at the ILC Symposium, including Maddie. Sydney just has only been here 2 months, so we give her a break on having a poster. And I will have a brief talk in the Lee Pate Memorial Symposium. So I don't get to go in depth, but I'm really honored to be kind of the main scientist representing what she did in the field. And then almost everything we do is on the website. If you want to learn more about what we're trying to do, we also have Front Range ILC has a newsletter, and I'm behind in updating it, but we've had a few, but it's based at this advocacy page. And if you are curious to learn more about the Lobular Breast Cancer Research Fund, this is the only plug I'll make. Like with the way that Lee and Laurie set it up, any dollar that goes to that fund comes to me, goes to research, like full stop. There's no other thing that fund is for. So if you want to talk more about how that fund support research, what we're doing with it, I'm happy to, but you can learn more at that site at the webpage. All right, so real quick, what is basic science? Like that is really the focus of most of these conferences. So what are we actually talking about when we say basic research, basic science? So this paradigm of basic cancer research is what develops ideas that we then have to take through translational studies, go into clinical studies that will change clinical care. It all has to start back in the lab and asking fundamental questions about biology. Where that starts, I alluded to kind of some model systems. In most cancer research, we're using different forms of model systems to mimic cancer in the lab. We can't just take all of you and do clinical trials and shoot random drugs into you. Apparently, that's unethical. So we have to do reductionist systems to ask those kind of questions. What that often looks like, and when we're done today, I'm happy to take folks over to the lab if anybody wants to hang around and see what it looks like in the lab. Should be quiet today, but I can show you some of these things. We have a series of good call. A series of incubators that are just kept at body temperature in human environments and allow cancer cells to grow in these effectively modified petri dishes. And these are cells that were taken from patients' tumors, some back in the 60s and 70s, and they've just been growing since because these cancers are mortal. And we can let them grow in those petri dishes and learn more about them. If we take this, these are literally shots from this microscope in the lab, and here's two different of these cell lines. Both of these have been growing since the 70s. This top one is from a ductal cancer patient, and this bottom one is from a lobular cancer patient. And I think one of the biggest things is that all of you, despite not being cancer research, can appreciate is that these do not look like these. And what we're learning is that the way these lobular cells look in culture actually mimics important properties about the way they grow in the breast. And we're trying to leverage that in the lab. But with these models, this is how we test new ideas, because we can abuse the daylights out of these cells because they're cancer. We want to kill them, right? We can treat them with different drugs. I can take tamoxifen. We can model aromatase inhibitors. We can try new therapeutics and see how the cells respond. We can do specific genetic manipulations to take certain genes in the cancer, turn them on and off using experimental systems. We can break the genome with newer technologies. We can actually edit the genome to look like the mutations that would happen in a patient. We can put that into a cell and see how it changes biology. We can manipulate the environment. We can change the fuels they have. We can change the growth signals that they get. We can change their oxygen environment. We can change kind of the 3D environment they grow in. We can put them something that looks like a breast. And with all of these different systems, we're asking fundamental questions about what makes these cells grow and multiply. How do they adapt to resist therapies? Ultimately, cancer therapy resistance is a problem, so we can see how the cells do that in the lab. We can try to learn more about how they move and metastasize, and we can try to see what makes them different from normal cells, because if we learn what makes them different, we can hopefully target it and kill them. This is just kind of the most reductionist model. We can also do things, there are advanced versions of these models that look more like tumors. We can also put fragments of tumor tissues into mice. One of the more recent kind of popular techniques are called patient-derived xenografts, where actually fragments of patient tumor tissue go directly into an immunocompromised mouse, so the immune system doesn't fight it. That tumor will then grow like a breast tumor, and then we can treat the mouse as like an avatar of the patient and see how the tumor responds to different drugs, or study other things about the tumor. And then ultimately what we want to do is take any findings we have and try to validate that with clinical samples. All of you, when you had surgery, if you consent to biobanking, your tumor is fixed in a block of wax somewhere in the black hole over there. And ideally, I ask to pull those things out, and then we have questions we develop in the lab, and we can ask, do these same things happen in patient tumors? And we have to identify different ways to measure that, to ask those questions. Ultimately, our goal, though, is to develop new diagnostic biomarkers therapies based on these reductionist models, and then move those through what's called translational research to translate the laboratory findings to the clinic. So in that space, obviously, it's not just do science, make clinic happen. Like, there's a lot of steps in between. So in the context of the conference, I just want to walk through quickly what the research life cycle kind of looks like. So first, someone like me or someone in the lab gets an idea, and then I have to beg for money to do the experiments. So we get funding, and then we do the research. And this is just some snapshots from the lab where we do some of this work, and I'm happy to take folks on a tour later today too. At some point, and this is where kind of today comes in, we want to present the research. And this is before it gets formally published. And this is where we want to talk about our research in a public forum to get feedback, to get ideas, et cetera. Inevitably, these presentations lead to new ideas, lead to new research. I have to get more money to ask different questions. so on and so forth. And then eventually we publish the research in an academic journal, and that kind of exists as the final version of record. The research is undoubtedly not done, but at some point we have that kind of the peer reviewable discussed version of record. Just to give you a sense of the time frame, this is for one of the kind of the major projects in my lab and how the time frame worked. So I started here in August 2016, and when I started the lab, I had some ideas I wanted to ask about estrogen receptor, and which we'll talk more about ER later, but that's one of the major proteins we think about in breast cancer as far as affecting biology. I had some questions about how do partners of the estrogen receptor affect ILC biology. Okay, so I had this idea in the fall. There happened to be a funding program internally that I was able to apply for, and I got some money in January 2017 to ask some of these questions. We did some research, and that, of course, leads to more questions. I have to request more funding, and that led to a few more grants. So we're already a few years in acquiring funding for the project. During this time, I'm not keeping our research in a vacuum. I'm talking about it as much as I can to other scientists to figure out, because I'm always trying to break new ground. We're not staying in our comfort zones. If I'm asking a new question, we're trying to ask other experts about their take on our data, how to interpret it, how to move forward. So I presented the research. This is a bunch of the different meetings. Some of this got presented at one of the virtual Lobular meetings post-COVID, always talking about what we're doing. Eventually, there is a version of record. It is a long process to submit, revise, etc. But ultimately, this started as an idea and a grant in fall 2016 and wasn't formally published until May 2021. So from funding to first publication is over 4 years. And that's not for lack of trying. That is just the reality of how research works and getting it to a robust set of conclusions that we believe in on the data. So that's, again, I can't say enough how important it is for us to present our data at conferences to get feedback, because if I waited 4 years to get any feedback on this project, inevitably it would not be the most robust, useful piece of data to the community that they need, that we all need to move the field forward. Yeah.
I don't know if we're going to speak to this, but I wonder if you could maybe just say a few words about the amount of money it takes in order to do this, because I think for a lot of people, you think, oh, $30,000. Money, right? But maybe could you just speak to that a little bit?
I wish I had the slides in here. I have a whole talk on research logistics. If you're really curious, it's linked through my website. There's an outreach and press page. I did a whole talk with Lobular Breast Cancer Alliance about the reality of how much this stuff costs. So for context, do you think anyone in my lab only makes $30,000 for a year? No, I mean, we're not paid that well in academia, but we do make more than that. So this kind of funding that starts a project is purely just for supplies. And this will last about a year for a small scale project. In reality, the funding, this was our mainline funding until recently, an American Cancer Society grant that was about $700,000 over 4 years. And that was a mainline funding. The project is now funded through a mainline federal grant that is about $1.25 million. And that's a very small version of those grants. We just got a new one to study some of this biology in ovarian cancer. That grant is closer to $3 million in total. So that's the scale we're talking about here. That said, I can't say enough about how these type of pilots are important in getting these ideas going, because even though I needed, frankly, $1 million to get this project all the way through and to give it momentum to keep going, I couldn't have done it without what was essentially a high-risk project that started with a smaller amount of money. There's stuff I can't do without those risky funds. So all of those numbers play a role. But yeah, I'm happy to kind of talk more about the nuts and bolts, and I have a whole talk on it. Thank you. Please, yeah, feel free to ask, especially when I get to biology. This is meant to be interactive too. So presenting at conferences, this is just kind of 2 examples of the scale and what this looks like. I mentioned Maggie in our lab presented at a Gordon Research Conference. These are typically between 80 and 150 people that are super focused on thought leaders. PIs will bring a few of their people in their lab, but it's hyper focused on a certain topic, and it's really meant to beat that topic to death. So the talks are small, they're very collegial. The poster sessions usually involve a glass of wine or 2 to keep the science moving. This is in contrast to things like the San Antonio Breast Cancer Research Conference, which is the biggest breast cancer research conference in the country. Over 10,000 attendees. This is the plenary hall. This is ridiculous. This is the poster hall, Exhibit A, Exhibit B. Bigger meetings that are not niche, because breast cancer meeting is still a niche. Society for Neuroscience is pushing 30,000. I don't know how big ASM gets. But like experimental biology is 25,000, AACR, American Association for Cancer Research annual meeting is 25,000. That's the scope we're talking here. The Lobular meeting is on this end of things. I think current registrants, including all our patient advocates, is between 150 or 200. Is that what we're at?
Yeah.
Okay. So much on the approachable end of things. That said, a lot of our patient advocates do go to San Antonio and meetings like that because there is science that's covered. There are people there that don't go to the other meetings. So they all serve a place. So real quick, why do we go to conferences? Like I just alluded to, a lot of it's about sharing ideas, getting feedback, and developing our own projects. So we're always excited to share ideas. Of course, there are scientists who don't believe in that, but I obviously do. We're trying to get inspired by new ideas. I have my ways of thinking. I've been trained one particular way. My background is all in estrogen receptor, how it works and how it responds to hormones and therapies. And sometimes I get myopic views on things. That's why I bring in people that have other perspectives in my lab, and I want those things at meetings too. We're always networking for new collaborations. At the most recent Gordon conference, we've started now regular meetings with Suzanne Konzen at Chicago, who does other hormone receptors in lobular breast cancer. And we're trying to now, just because I've talked to her more at meetings, we're getting more relationships going and interacting more. We're always trying to find career development opportunities, both to recruit people to the lab and to identify the next steps for our trainees. Where are they going to go next? I got this job in Colorado because of a meeting, a Gordon conference. That same Gordon conference I was talking about, I didn't shut my mouth there a lot, and that ended up getting me a job here. So it worked out okay. Sometimes it's important to take a step back for us from grant writing, from the day-to-day grind, and go to a meeting and get a chance to just think big picture for a while. Not unlike any other fields retreats. If we're trying to go into a new direction, it's easier to learn about a new field. We can read papers until our eyes bleed, but sometimes it's just easier to learn from the experts directly when they talk about new ideas. And we're often trying to broaden horizons, whether that's scientists learning from physicians, learning from patient advocates, and vice versa. Conferences are great for that. Why do patients and advocates go? This is my last slide for now. This is just a few snippets, one snippet from GRASP, which is another one of these patient advocacy and outreach groups. I'd highly recommend you to look into GRASP if you're interested more in kind of this researcher scientist interface. But obviously, advocates go to learn about what research is being done in their space or more broadly in the community. Teaching is the more important thing. What Maddie alluded to is that it can be hard for a lot of scientists to remember why we do what we do. And then it helps us reframe the questions we're asking in the lab when we think about what's important. Like, I can kill a cancer cell in the lab really easily, but if it takes 4 drugs to do it that would kill a patient, it's not a very helpful line of research. And sometimes we have to think about those things. Finally, in terms of advocacy and awareness, Congress cares very little about what scientists yap about. Like, I go sometimes to the Hill to advocate for funding in congressional offices, but for stuff to actually get done, it takes patience. So if you know what we're trying to accomplish and we know what you need, advocates can always be better amplifiers for the needs of their community than scientists can. It's just the nature of the beast. All right, that. Any questions about conference, the life cycle? All right, we can always come back to this stuff, but I'm really happy to hand it off to Devi and Laurie and Project Bridge to talk about communication at a conference and a poster session. There's no video, it's just the mic on us.
Yeah, so we said this earlier, we're really excited to be here. Did the introductions already, but just like Matt said, we want this to kind of be interactive. So if you have any questions at any point, you don't even have to raise your hand, just speak out. We're more than happy to talk. If we don't cover anything, ask those questions. So that way, hopefully the next time we do this, we'll be able to improve upon it.
Yes, and before we get started, I just want to gauge how many of you are planning to attend the ILC conference, whether that's in person or virtually? Okay, fantastic. Yes. Okay, so we have a couple here. And so hopefully some of this information will be useful for the rest of you for the future, getting more involved and attending scientific conferences. Maybe, hopefully our goal is to make this less intimidating for you, so you're more excited to jump into those spaces.
I'll say there are more than happening on campus. There are also about mediation advocates for attending the poster sessions. So whether it's our group or others on campus, it's getting momentum. We'll have more opportunities for this.
Fantastic.
Cool. So again, I briefly mentioned this earlier, but we are a chapter of the National Project Bridge organization. We just became affiliated officially with the university last year, although we've been on campus since 2017. Yeah, I think that's right. So yeah, our main goal is to try and bridge that gap between scientists and their communities, hold workshops like these to either train scientists how to communicate better or get the public engaged in scientific research. So we have a couple pictures up here. The top two are actually from an event we had in January of this year, where we had 50 scientists from across Colorado, a whole bunch of different disciplines. They were able to present their research at the Capitol to legislators and the public. And so we had to do a lot of training, because we'll be showing you here in a little bit what scientific posters look like. That's not really helpful for people who don't exist in that space. So we had a couple of workshops on how to take what is a traditional scientific poster and make it easily accessible for the general public and for legislators. And that was really awesome, and we're planning a second event this next year that will hopefully be in March. So that's the brief overview of what we do.
And then an overview of what we're planning on talking about today. We want to talk about what is a poster session, what is the purpose of them, and who you might see there. Like Matt alluded to, how scientists are taught to communicate versus what the public is actually looking to understand, and then how you would interpret a scientific poster. So we have some example posters going over the general anatomy of a poster, and then I'll talk for a bit about just communication at a scientific conference in general. We've realized doing this that there is a lot of weird social constructs that we've had to get used to over the years. And so we just wanted to take all of the things that we've learned through experience and give all of those tools to you. And then the last thing, just trying to reach out with someone after a conference or after you've had some type of interaction with a scientist. What do you do afterwards? How do you follow up with that? So that's generally what we're going to be going through.
And just briefly with that, reaching out after conference, if you're unable to attend the conference next week, I think a lot of the posters will be publicly available afterwards. Matt's nodding. So yes, so even if you aren't able to go, you should be able to have that access to those posters. So hopefully the information that we're giving you today, you can at your leisure peruse the posters and hopefully get the information that you want to get out of those using the tools that we present. So Matt, again, already kind of mentioned this, but why do we have posters at conferences? A couple of different reasons. Mainly it's for early career scientists to present their research. You have a lot of speakers that also present at these conferences, but they tend to be more late stage scientists like Matt. So experienced. So it's predominantly early career scientists make up the majority of those that are presenting at these poster sessions. And a lot of it is to receive feedback so that they want to be able to talk about the research that they're doing because they're excited about it. They put all this effort and work into it, but they also want to get feedback on it. What next step should I take? Oh, I didn't think about that question. I should look into that. And then it also facilitates networking. So like Laurie and I are nearing the end stage, hopefully, of our PhDs. Laurie's looking into new postdocs. Being able to, as Matt talked about, interact with people that are doing similar research to you is really helpful for trying to figure out those next stages for these early career scientists. So generally who you can expect to see presenting is graduate students like Laurie and I, postdoc researchers, some PIs do present. You also have clinicians that can be presenting, and a lot of times you'll have technicians like Maddie and Sydney that will be at these poster sessions. So how scientists are taught to present their research differs quite substantially from how the general public wants to consume scientific material. So this also is pretty closely aligned to how we actually carry out the science and the experiments and the research that we do in the lab. So generally speaking, we want to have some sort of background and introduction. You need to know a lot about what you're researching in order to do the research. So we do a lot of reading, talking with people to get that big overview and background information. Then after you've figured out the questions that you want to ask based on that information, you then go into figuring out how you're going to ask those questions. What experiments am I going to do? What reagents do I need? What cells am I going to grow? Then you do all of those experiments, you get the results, you put it all together in pretty graphs that end up on these posters, and then you form your conclusions from those results. So in general, this is how we are taught to carry out these this research, but it's also how we present posters. So little cartoon on the right is the general schematic of what you're going to see and how a poster is presented and laid out. It can vary, but generally this is what you're going to see. So that background and introduction is mainly going to be found in that abstract and introduction side, usually on the left, usually at the beginning. Then I'll go into the nitty-gritty details of the methods, followed by the results sections of the experiments that were carried out. And then finally, the discussion usually includes that conclusion, next steps, why do we care? But that is not how the general public wants to consume information. It's a lot of information. Unless you're living and breathing that science, a lot of this is not helpful, at least initially. So non-scientific audiences really want to know initially, what is the big picture? What are the results? This drug does work, or this drug doesn't work. You want to know that first and foremost. Then you can start getting into the details of, okay, so why do we care? What are the more detailed information? And then finally, you can get into the really nitty-gritty details. But you're not going to get to that supporting information unless we talk about the results and big picture first, because we're very busy people. We have other things we want to focus on. So,
And clearly you as an advocate, you're less concerned with the rationale because most of you are the rationale for the experiment, for all these experiments. And you want to know how this research is going to impact you and your community and the people that you care about. So Devi will tell you how you're actually going to get this information that you want out of these posters.
So we're going to go through this poster, cartoon, and talk about what to look at these different stages. So the poster title, look for keywords. So if you're interested in different like HER2 positive, ER positive, all of these different things, hopefully you'll be able to get some keywords out of the poster title. And that should help you decide whether or not this poster is worth your time and effort to get more information out of, because there's going to be a ton. You can't go through all these posters. You can't attend everything. So you have to pick and choose which ones are going to give you the most information for what you're looking for. Then you want to look at who is presenting. Laurie's going to get into this a little later about how to actually engage with people at a poster session. But generally speaking, you're going to have a whole list of authors at the top. The first author is generally the person who is actually presenting the poster and is the one who's doing the primary research, like the hands-on. There'll probably be a couple of other authors that are listed that were involved, but then the last author is usually the principal investigator, like Matt, who's wrote the grant that got funded and whose lab in which the research is done. So we would really suggest that after getting the title and getting some keywords, look at the abstract. Don't get bogged down in like the results section. Really just look at that abstract. It should be a very brief summary of what they were doing, why they were doing, and the conclusions. So it should summarize everything nicely. If a poster is a good poster, at least in our opinion, the acronyms that you might see throughout the rest, they get very detailed. Like if prior to diagnosis or hearing about invasive lobular carcinoma, you probably didn't know what ILC stood for. So hopefully you'll be able to see these acronyms in the abstract that will help you understand what the rest of the poster is talking about. And this is really awesome for this conference. I have not attended another conference that does this, so this is great. This conference will actually have a lay abstract. So they'll have their regular abstract, but also a lay abstract that's for the general public. So it hopefully will make it easier for you to understand. Yes.
Unfortunately, those are being written by scientists.
Yes.
Related, so I've been to other conferences where they actually have a lay summary written by lay people with scientific background.
I love that.
Scientists can edit it and say, oh, you totally missed the ball, or yeah, you got this in the but sometimes lay abstracts written by scientists still read like scientific abstracts.
Yes, very much so. And I will just have another plug for Project Bridge and the importance of organizations like this, right? And hopefully we can train the next generation of scientists to be better at communicating their own science. So you don't have to have these go-betweens. They are able to effectively communicate to the people that matter. So.
And I'm glad you brought that up because I will be talking about how to ask questions later when it's not clear. Even when they're trying to make it clear, it's still not. So thank you for that.
And so finally, after looking at the abstract, I would have a brief look at that discussion, which should contain that main conclusion. So that big you know, what's the main conclusion? Why do we care? Hopefully is found in that discussion if it was briefly mentioned in the abstract. It might also include what we consider to be a model figure. I'm a very visual person, and so if they can like show me a cartoon of what they think is happening, that helps my brain figure out what exactly is going on. I like a lot of posters and papers that have these model figures. Hopefully a lot of these posters will have something similar in the discussion that kind of gives you an overview and a visual representation of what's occurring. And it can also include next steps. So like, okay, we found these conclusions or we got these results. Here's what we want to do next, or here are other questions that we have based on those results. So hopefully based off of all of this information, looking at the title, the abstract, and the discussion, you can decide whether or not the poster is essentially worth your time, right? Because you're busy, you have other things, there might be other posters that would be more beneficial for you to look at. So at this point, you can hopefully decide if it's worth your time, or you can move on to another poster. And so we wanted to practice that a little bit. So we have some example posters that Matt was able to provide. So this is one of them. It's kind of hard to see.
It's a good thing though, because we don't want you guys to focus on these results sections anyway. They're a bit overwhelming, and I think they're more helpful for when the scientists are talking to other scientists at their poster and getting that critical feedback that Matt was talking about earlier. So you can take it forward, but
yeah,
the parts are larger that we actually want you guys to
which
try
is
to
nice,
see.
which is helpful. So yeah, so
So this poster is actually from Range on the OBCA Scientific Advisory Board. This is from San Antonio last year. I just pulled up Twitter.
Yes, it was from December 2022. It was, yes, I pulled what you said off of Twitter.
So this is a relevant poster.
So we kind of want to go through this, and we wanted everybody to take a look at that title. And again, I'm hoping to be more interactive. So if you see any keywords that pop out to you, we would love it if people could say things. You can't hear what I'm saying? Thank you. I'll speak louder. So we were hoping that people could read the title and say any keywords that pop out to them that would make them interested or not interested in pursuing the rest of the poster.
Also, if you can't see it well, the title is Self-Identified Race and Area Deprivation Index in Patients with Invasive Lobular Carcinoma of the Breast, Associations with Tumor Characteristics and Event-Free Survival. I did not know what many of those words meant when I read the title of this poster, so it's okay if you don't either, because I'm just a scientist in a different field. But was there any words in there that piqued your interest that might make you want to go to this poster?
I'm kind of curious about the event-free survival.
Yeah.
Event-free survival, fantastic. So I didn't know what that meant either. I still am not sure I know what that means, but that was something that caught my attention as well. Where else? I heard someone over in this area.
Same thing.
Okay.
I know when I was reading it, I got area deprivation index. That sounds important. I don't know what it means, but it sounds important, right? So there are a couple of keywords that like we might not know exactly what they are, but we can kind of key into them. Now, thankfully, this poster is awesome because if you look at the introduction on the left, it actually explains a decent amount of what those potentially jargony words are. So you can see under research question, they have area deprivation index, which is a robust indicator of socioeconomic status. So that explains a little bit better what they mean by this area deprivation index. And as we mentioned earlier, they talk about event-free survival, which later on they refer to as EFS. If they hadn't explained what EFS was, we would have no clue. So it's really nice that they included that here in the introduction. This is also unique to this poster that they have a key point. This is great. This is like that big question, why do we care? And they have it bold and right in the center. So I think this is great because it grabs people's attention. On the flip side, if you are not very familiar with this, the key point being area deprivation index and body mass index mitigate the relationship between self-identified race and DFS in patients with early stage ILC, that can still be a little jargony, right?
I still don't know what that means.
But you can hopefully pick up some key points, and if there are parts that are at least interesting, you can then go on to ask pointed questions to the individual that's presenting, and Laurie's gonna get into that later. So we kind of wanted to ask if there was anything that made you feel comfortable about this poster versus uncomfortable? Or that made you like want to ask more questions, or that made you kind of like maybe want to back away slowly? Yes.
Well, I don't see the abstract, which is where I would go when I look at these studies, and that's about all I can handle. As soon as I get into the charts, and I often can't understand. And the conclusion also can't see the conclusion, but that's where I would go, is to these two places to see if that's information that's relevant for me or not. And often, you know, the abstract will answer the questions.
Yes.
So, but this one doesn't have an abstract as such, right?
Yes. So
I agree with that, yeah.
So the abstract is kind of shoved into this introduction and research question.
Okay.
Is that area that I would look for. So if it doesn't have a clearly defined abstract, I would look for that introduction section. They can get a little jargony, but hopefully that introduction section will have that background information, why they're asking the question, maybe a little bit of what they did, and hopefully some conclusions. So
Yes.
So I would say, so I'm pretty familiar with scientific posters, but not from the medical side of things. And so immediately when I look at this, I see the white space, which is super inviting, because so many times, like, I know that they're like, oh, this is important, I need to include this too, and it becomes so overwhelming. So like, if I was walking through a poster hall, I would probably stop at this one just because it looks tractable. Like I could actually maybe understand what's going on. So.
yeah, that's a
We do have one of yours coming up next, and it is less white space, but I do see what you're saying. I gravitate toward those posters as well. It is.
It's just that if it's in an area that I'm not as familiar with, right?
Right.
Like if it's an area that I'm familiar with and I feel comfortable that, you know, something like this, which is outside of my expertise, then go for the low content.
My comment would all actually be opposite of that, because the two graphs that are almost in the conclusion area are so light, and the text is so small that you're not going to be able to read it unless you're standing right there in the poster. And compared to the areas that are brightly colored, you're not looking at the plot over there that says what your answers are.
And I guess to add on to that, I'm also thinking that I'm gonna talk to the researcher who's standing there at the poster. But I agree with you that if I was just walking by and there's nobody there, I probably would say, oh, font's a little small.
So I think this is, I think this is something we struggle with is, are we making the poster so that someone stops and talks to us? Or are we making the poster as a standalone unit information? So a lot of us don't include the abstracts anymore, because the goal is to get people to stop and talk to us. But you're right, after the fact, then there's a lot of information that's not there. So it's something we struggle with.
So like me, if I was to see it's the early stage ILC, because that relates to us, right? So at a meeting, we can see my interest, but then I'm interested about the BMI relation to the deprivation, you know, index. So now I want to know a little bit more about it and see.
Well, this is all great, and I'll say thank you all, because we will be taking a lot of your comments into consideration for the next STEM poster day when we're trying to train scientists to make engaging, inviting posters. This is all really, really helpful information. But yeah, I agree with the colors, I think, stand out, the color palette that they went with. I think the writing, they don't have, it's not all of these words crammed together in like long paragraphs without these spaces that just makes you tired looking at it, right? And I will say, in terms of a lot of the graphs, there's a few, but it's pretty sparse. When you go to these conferences, people are very excited about the science that they're doing, and they want to show you every little thing they've done. And so they try and cram as much as they can in, because, you know, they put a lot of work into it, and the results that they get are important. But we as scientists need to make sure that we're being better about how we're communicating that, specifically what is the main takeaway and how do we get people to engage.
And one of the things I liked and the thing I wanted with this poster was, like Devi brought up earlier, a model figure. Like I would love to see a picture that shows what their conclusions were, because I, being unfamiliar in this field, could not gauge what their key point is. I didn't know what mitigating the relationship between meant. What does that mean for cancer patients? What did that mean for this relationship? That's something I would have asked the presenter, but I did like, they did over here their methods with just a picture, and it says electronic medical records, patient characteristics, and address and area deprivation index. Two things that they got, two pieces of information they used to conduct their study. That made sense to me. And it's very small, I can probably not read it on this, but with a higher resolution, probably be able to read it. But I love things like that. Just give me arrows and pictures. That's what I want. Yes.
If there's not gonna be a lay abstract or a poster, then the key point is actually hugely helpful.
Mhm.
And right after the title, that's where my eyes go to tell me,
Yep.
I care to read more about this, even if I don't quite understand every nuance of the key point, just having that there is huge.
Uh-huh.
That tells me, yeah, I wanna learn more about this, or this is not necessarily of interest to me, or I have a question about your key point. And your conclusions are so small that without that point,
Yeah.
it's almost hard to discern what it is. And so I love that they kind of tell you right up front, here's what we found. If you want to learn more, you can read the fine print.
Yeah, exactly. Gives you a jumping off point, like an anchor point. And I will say a lot of posters don't have the key point, which is disappointing. Hopefully, as a field, scientists will move towards these sort of layouts to help communicate better. But yeah, I agree. It's a really, really good place to look at title, introduction / abstract, or key point.
Can you read that conclusion? I can't see it.
Gosh. Yeah, let's see. I'm not even sure if I can. Let's see if I can read it on here.
Univariate analysis.
Self-identified Black race was associated with worse outcomes in stage 1 through 3 ILC. However, this is mitigated by adjusting for area deprivation index, which is strongly associated with both self-identified race and body mass index.
Yes.
In English? So any cancer patient, the outcomes tend to be worse for non-white patients, in particular Black patients, which is what the focus is here. What this analysis is saying is that the poorer outcomes for those patients are not because they're Black, it's because they can instead explain it with this area deprivation index, which is a measure of socioeconomic stress, and BMI, which is weight. So they're saying that these patients have poor outcomes, but it's not because of them. It's because that happens. Being Black is associated with these other things that are also bad.
And the social worker in me, who reads stuff like this goes, well, don't a large portion of Black people tend to live in places that have low ADI and very south, higher BMI, so maybe it does have something to do with that. So it's a little, you know,
Yeah. And that's fantastic. As someone from Louisiana, I really appreciate that these studies are being done because that's a large portion of our population. So
Food and medicine.
very true.
The whole state almost is the
Yeah, that's very true. Very true. We should talk later.
Let's do it.
Awesome.
All right, move to the next one.
Yeah. All right. So this is from Matt's lab. Oh, it's so fuzzy. I'm sorry.
Yeah.
So yeah, the screen is not, it looks great on our computer, but less so up here.
We do have another poster from that slide back there. If you guys want another practice later, that one you should be able, you can get up close and read like you would be able to in a poster session.
But this is, I think what like a more like typical poster will look like. So you have that, except it includes that lay abstract that we talked about. The ILC conference will include that lay abstract. So you have that, you can look at the title again to look at those keywords. Read that lay abstract that hopefully does a better job of explaining the science than the traditional abstract would for those who are not well versed in the subject. And then you can again look at the conclusions and then decide from there if this is a conversation that you want to have, engage with the presenter before getting into all of the really pretty graphs and colors that you see in the middle.
So what's PARP?
Yes.
PARP inhibitor.
that one
Palazoparib. I've heard that one before. But that would be considered a keyword, right?
Yeah, Matt, what is it?
No, you're fine. I just need to shout. Okay, so no, it's fine.
Abstract to us, just so we can get an idea, like, all right, so like, oh, I'm here positive ILC, great, that's me.
Yes, I can read the one down here. Despite being typically cast as a low-risk breast cancer, invasive lobular carcinoma is linked to poor long-term outcomes and resistant to anti-estrogens, which is complicated by a lack of ILC-tailored treatments. Our research identified a potential treatment vulnerability in ILC cells related to the ability of cells to repair damage to their DNA. We found ILC cells have a defect in their DNA repair capacity, which can be exploited using FDA-approved PARP inhibitors like Talazapar. So that one. We tested whether ILC cells are sensitive to talazoparib, excellent, compared to invasive ductal carcinoma cells. ILC cells were more sensitive to than IDC cells and less able to survive treatment. However, some anti-estrogen resistant ILC cells were PARP inhibitor resistant, related to changes in DNA repair. Our work identifies PARP inhibitors as a potential treatment for ILC and will be explored in tumor models with paired biomarker studies for DNA repair activity. I thought that one was pretty good. I like that lay abstract.
So just kind of a little reminder of just the differences between how scientists are taught to present and taught to do science versus how the public wants to consume that information. It's very different, essentially opposite of one another. So just when you're looking at these posters, reading them online, talking with the scientists in person, hopefully the scientists will do a good job of explaining, but also remember that that's not currently how they are trained to present that information. Cool. So now Laurie's going to talk. Okay, cool. We've given you some tools on how to like read these posters, but how do you interact with the scientists and how do you get more information out of these posters once you've decided it's worth your time?
And so I didn't bring this up before, but this is almost a cheat code to a poster session. This is my best friend whenever I'm at a conference, is usually there is an abstract booklet. So apparently this one is not published online yet, or it won't be, but at a lot of conferences, you'll have an abstract booklet that will give you all of this information. So it will say, usually the location of the poster. I pulled this from a different conference, so this was not from ILC, but you'll get the title that you can look over, authors and affiliations, so you might know who you're expecting to see at this poster, and they'll give their background, their research question and hypothesis, what were their methods and their results. So I will often scan these abstract booklets and star posters that I'm interested in, and you can read that information and kind of start to formulate some of your questions before you even see the poster at all. If you're looking at these things virtually during the conference, like I mentioned, you can view posters, then you can peruse at your own leisure. But sometimes it's hard when someone's standing there at their poster, so ready to talk to you, and you're just looking at it for the first time, trying to digest the title, and they already want to talk to you. So this can be a great way beforehand, or even if there's just a list of titles in some type of booklet that says who's presenting posters at this conference. Even just the titles, if they don't give you all the information, you can start to decide. Maybe at an ILC conference, you're interested in more of the posters, but for example, I'm a microbiologist. That's bacteria and viruses and fungi. But I look through, and if I see anything that talks about women's health and the female reproductive tract, that's a star. Anything that's talking about the bacteria I study, that's a star. So it will help you to narrow down when there's tons and tons of posters. What do you want to actually spend your time on? Because your time is so valuable at these conferences and you want to make the most of it. So that was just my plug for the ways you can possibly prepare yourself beforehand at conferences in general. And so once you've selected some posters you want to talk to, how do you actually go towards approaching a poster at a poster session? And so like I mentioned before, if it's virtual or you're at a poster session and the person is not present, oftentimes at conferences, the posters will be set up in a room before the actual poster session, and you might have time that you're allowed to walk through, but the presenters won't be standing there at their poster. And that can be a good time just to peruse at your leisure. Maybe if you don't have an abstract booklet, you can make a note of, come back to poster 502. This looked interesting. So spend some time looking over them, write down any questions you might have, or if you know you won't make it back to that poster, possibly, you can always write down the contact information of the poster presenter. And we also have a few pictures of when presenters are at their posters and you get to have an engaging conversation. We also have Devi and little Sanji there, so cute. Baby's first poster session.
Baby's first poster session. I thought it would bring people in more, you know?
And it's also a reminder too that the people who are presenting these posters are people too. Like they're scientists excited about their research, but they're people just like you. And they really want to talk about their research. And I think one of the most important things for you guys to not immediately get overwhelmed is in approaching a poster, introduce yourself. Say, hi, I'm a patient advocate and this interested me about your poster. And hopefully in the presenter's mind, that will kind of, I mean, they haven't seen our beautiful triangle diagrams, but say, this person doesn't want to hear about the statistics on my research graph. They might want to hear more about the big picture message. And so it's helpful too to lead them in and say, this is what interested me about your poster. Just a great way. They have a million things going on in their head. They're tailoring to many different audiences. So helping them zone in on what they should talk about with you will make the most out of both of your time. And just from my own experience presenting posters, there's kind of two main options or ways that this usually goes. Either one, if you just want to know more about this project, you don't have a specific question necessarily. I often don't have a specific question when I want to hear about a poster. You can just ask them for a quick overview of their poster. Even saying, could you give me a quick 3-minute overview of your poster? And that can clue them in on, I want to talk about the most important things and just hit the highlights. And then you might be able to develop questions you might have after that, or you can always say, thank you so much for your time, great research, and move on. So you don't have to be stuck in a conversation. And then option 2, ask a specific question. If there's something you are really interested in or want to know more about, ask them. And while they're talking, you want a poster presentation to be a conversation because they're not giving a talk. At talks, you're not interrupting, but this is a poster session. So interrupt politely. And obviously this is, I'm sure most of you guys know how to do this, but it's more so if they're coming to the conclusion of a section, you can always jump in and say, okay, so, or I heard, so I have a section on all of this later, like how to ask questions and how to interact and listen at a poster. But keep in mind that you talking to the person at this poster is a conversation. Some people like to just give it as a presentation, but feel free to interrupt them if you have questions or they're moving too fast or you're not understanding exactly what they're saying. Please ask for clarification because scientists often don't know when they're not being clear.
And I will say, do exactly what you have done today. Like how you have be like, I'm sorry, that didn't make sense, or can you give more detail about this, or I saw that. Exactly what you have been doing. Do that at the poster session.
Yes.
Yeah.
It will be posted. And like I mentioned before, it's okay to not engage with the poster presenter. If you are scanning the posters and the person standing there really eagerly at their poster, I will say sometimes to people, if I see them looking at my poster, do you want to hear about this or are you just looking? Some people won't say that. Like, I'm just conscious of people who might not care about what I'm talking about. But you can say, oh, I'm just looking, or, oh, can I just have a minute to look this over? And it's fine to walk away after that, or just say thank you, or like, great poster, and move on. That's totally fine. This is a session with many different people. Someone else will come up and talk to them. You don't have to feel pressure if you're like, this isn't really my jam. So that's totally fine too. It's these weird little etiquette things that we've picked up over the years that for us, even as poster presenters, can be really awkward and uncomfortable meeting strangers and talking about our research. So
no one explained it to us. Like we just kind of had to, it was like, okay, poster session, go, and trying to figure out these somewhat awkward social nuances that you don't know right away. So hopefully this eases it for you all.
The first time I'm itching to talk about my research and someone's just looking at my poster like this. And you get, oh, hi, and you get a hand. I was like, oh no. But
We're very.
You know?
I feel like I'm a very extroverted scientist. I feel very awkward, and there's many more introverted people than me. So so this is generally approaching at a poster session. I'll go over in a couple slides what that conversation could actually look like. But you may also, if you're attending a conference, come to a talk. And like we mentioned, this is often people who are running labs, principal investigators, giving talks about research that's usually at a bit later stage. They might be talking about something that's already published. And when you're attending a talk, it can be really helpful to take notes. I honestly have to take notes or I'm not processing the information. But you can identify questions you might have at that time, and then whether they answered those questions or not later in their talk. And during a talk, there's two main times that you can go up and ask questions after a talk. One is usually a designated Q&A session. So after the talk is over, like we'll have at the end of our slides, a questions slide. And like down here, there's often, if it's a large conference, you can go up to a microphone and stand in a line, or someone will come around and give you a microphone to ask your question. And so that's a good time to ask a short question, introduce yourself. Hi, I'm so-and-so, I'm a patient advocate, and my question is, and try to keep the questions short in general, because everyone's going to be hearing the answer to this question. So this really isn't the best time to ask a really specific personal question. But outside of the designated Q&A time is a great time to have an in-depth conversation with a scientist or a researcher that you heard give a talk. And this is another one of those times that no one taught us how to go ask questions after a talk. I understood the etiquette of the designated walk up to the microphone questions, but this one can be really hard. And so one thing that we probably all understand, but it can be awkward, is being respectful of the presenter's time. They might be talking in a really big group of people, and it's probably not the best time to go in and interject. But waiting for a break in conversation or catch them when they're alone. I've often gotten approached at coffee breaks, and it's so exciting that someone says, hey, I heard that thing you said earlier and I like that, can we talk? So exciting. But trying to catch them immediately following the session, when they're leaving and they're not talking to anyone else yet, or they step away to grab coffee or something, great times to go up and interact. And even if they're busy at that moment saying, hey, catch me again later, they'll have that facial recognition. You can at least introduce yourself and get the point across that you want to talk. So that's a good time. And just an easy opener is, oh, I really enjoyed your talk during this session, just so they know you were present at their talk and that's what you want to ask them about. Or even if you see someone's poster, you're really interested in a poster and you see their name tag, you say, hey, I saw your poster and didn't get to catch you. Can I ask you some questions? So it's another time that it's really good to have notes written down of any questions you might have, because I always think that I'm going to remember the questions that are in my head, and it has not happened once that I truly do. But yeah, just introducing yourself again as a patient advocate to make sure they're tailoring their answer to you. And I think my favorite part of the talk is coming up, that we want to practice active listening. And I speed added all of this to the PowerPoint. Yes, yes.
For Q&A after a talk, I would say that one thing that is not appropriate to ask during the open Q&A is about personal breast cancer experiences, your own tumor, your diagnosis. That's one thing. That's something you can talk after the session, afterwards. Don't bring up any like specific diagnosis question during the meeting. Like physicians can't talk about it in specific cases anyway, and scientists definitely can't talk about it. So if you can ask those questions, like tailored answers about that after the session.
Excellent point. Fantastic. So when we're talking to a researcher at a conference or just to each other, active listening is a fantastic skill to practice. And a lot of us already do these types of things in our everyday life sometimes. But on the next slide, I'm going to bring up some skills that you can actively use while you're talking to someone at a conference. And active listening is basically what it says here. It's a communication technique that involves attentively processing and understanding the speaker's words. As well, like people can say words, but nonverbal cues are very important as well. You can tell when someone's uncomfortable when you're talking to them, and they're not saying I'm uncomfortable. You can see people back away. You can see people's demeanor change, and knowing how to look for those things and tailor your words and your actions. And it's also important just to demonstrate genuine interest and thoughtful responses to what people are saying. Helps make connections, deeper connections. People will remember their interactions with you. Because I always love the phrase that people won't remember what you said, but they'll remember how it made them feel. And I think that is a very important part of active listening, that they will remember that they enjoyed their conversation with you. And it can help you in this context get the most out of a poster session or just any type of informal interaction you're having at a conference or in your daily life. And so over here is just like some general, these are things that you're probably more familiar with, like face the person, eye contact. That's really important. Make sure you're not, check your posture, like don't look like you're angry or something, even if you're really angry about something else. And avoiding distractions, like when you're at a poster, I'm so bad about this. I want to check my phone so bad. And I just felt it vibrate in my pocket, but I'll step away from the poster or say like, if I'm in a big group of people at a poster, I'll still step away before checking my phone or things like that. And trying to engage.
I actually have an example of that. So when I first joined Matt's lab, I think I was there a couple of months and he brought me to the conference in LA. First poster, never presented a poster before. I've been in the lab a couple of months. I barely know what I'm talking about on the poster. So nervous, standing there, waiting for people to come by. One of the big wigs from the poster session comes by and he, stands there, looks it over. And again, I've never done this, so I have no clue social cues. So I'm just like, waiting for him to say something. And he looks somewhere in the middle of the poster and I can't remember what it was, but he asked, "What's that?" And like, it was like, I'm sorry, but I'm not sure. Like, I've only been here a couple of months, but I can, get back to like panicking, trying to figure out. And he just walks away. That was it. It's been 6 years. I still remember that interaction. It wasn't great. Don't be that person, right? Like, I was super excited to be there, nervous as heck, but like, talking about the research that I'm doing, the research that I did over the next 2 years, something I want to go back to, like it's something I was very passionate about. And it sucked to have someone who was, higher up in that field have that type of interaction with me. So just remember that, like Matt was saying earlier, we're just as scared talking to you guys as you are of us.
Scientists need to learn these skills as well. It's a PowerPoint for you guys, but many scientists very much need to learn some of these skills.
Yeah, it's mutual respect, right? So just keep that in mind when you're interacting.
And as has come up today already, the scientists care about your opinion so much because people with this diagnosis, that's who the research is for. Like they will care so much what you think about their poster and what you think about their research. So you guys are wielding a lot of power going into these spaces, honestly. So one point that I brought up before, be attentive. So we talked about this already. Eye contact, open posture, try to remain mentally engaged. I have trouble with this. If they're losing you, let them know that they're losing you. Try to stay engaged because they can probably tell when you've glazed over. And a good science communicator will be able to notice that you've kind of glazed over and they will pick up on that, but some people won't. They're just so excited and they're breezing through their rehearsed dialogue. And one of my favorite things, I can't listen without doing this, nodding along. I'm always nodding along. If I understand a point, that's a nod. Yes, that exactly. But it's helpful when I see people doing the same things I do, nodding along when they're talking. It can help a lot. And so number 2 is asking open-ended questions. So these are questions that don't have a simple yes or no answer. Something like, tell me more about this part. What do you think about this? Or help me understand. Help me understand could be a great tool to use. Those words, the area deprivation index, I didn't know what that meant. Help me understand, what does this mean? Like beyond the text you have, what does this mean? And then 3 will be less important for you guys. I think this is more for the scientists, because I think this comes a lot more in the results section, asking probing questions. But there could be parts of a topic that you're really interested in and you want more detail. These are your how, what, why questions. Why can be very helpful for you guys too. Why did you do this? Why did you do this certain way? Why did you choose this study population? How did you select this? Can be really helpful questions to ask. And I think a really important one for me when I go to posters, and especially for you, request clarification. It's okay. That is an active listening technique. ask them to clarify a point that doesn't make sense. It shows your interest and it ensures that you're receiving the correct message that they want you to receive. So this exact, I've used this so many times. When you say this phrase, like you keep saying this, does that mean this? Put it into your own words, which kind of goes with the next one, paraphrasing. Telling them how you're understanding what they're saying helps them figure out if that's actually what they meant. And that can really help scientists when they're giving their presentation to the next group of people. If they realize what they said wasn't clicking, they might just take what you said, being like, oh, that is a better way to explain what I'm talking about. I've done that frequently when presenting. So paraphrase kind of goes along with that. So if I understand you right, you mean this, or in other words, what you're saying is this treatment is working on cancer patients. Something that gives them a clue into what you're thinking.
Just like how Matt did earlier, where he summarized that poster for us before, we were like,
Yes.
what? It was like, oh, okay. It's not because of this, it's because of this. Like, that's a really good question to ask, and it helps them clearly communicate what they're trying to as well.
And then this one, I wasn't sure that this one was going to apply so much, but then I thought about it a little bit more. And what we were just saying, that your opinion means so much to these researchers. Be attuned to and reflect feelings. Looking at a poster and saying, I understand why you're so passionate about this research. This is really great. Or as a patient advocate, I appreciate the work that you're doing. That can mean so much to a researcher and help them push forward in their career. So I do feel like they're very much going to appreciate if you employed this skill. And then the last one kind of ties in with number 4 and 5 as well, but summarize. When you're finished talking at a poster and you're ready to leave, just saying something like, oh, okay, so I've like learned this, this, and this. You can also ask them about their next steps. So where's this research going? Like the questions from earlier. So is this at a point that it could move into clinical trials? What is the next step of this research? Like Matt's, okay, we're moving from cell line to mouse model. Like that's the next step in this research. When do you see this being published? Things like that, just general summarizing questions. You don't have to ask any of those things, but it can just help to wrap up the conversation and show that you were listening and you understood a little bit what they were talking about. So all of this will be in the presentation that you guys can see afterwards on YouTube. So if you want to practice any of these skills when you're going to a conference in the future, you can always reference it again.
Well, I'll make it 5.
Fantastic. And so we mentioned this before, but what if you just have questions that didn't get answered? What if you're not able to attend the conference and you looked at these posters or slides virtually online, but you have questions and there's things you want to know and you want to engage with these researchers? The best way to do this is by email. You might be able to search online and find people's LinkedIn's, their Facebook pages, a lot of different things, but reaching out by a professional email address is 100% the best way to go. So the first thing you want to do is identify who is the correct person to contact. Is it a graduate student researcher who's doing the poster? Is your question about the research they're doing? You might want to reach out to them individually, and you'll probably have better luck getting an answer from them, from the earlier career researchers, because the PIs have so much on their plate. They're writing all these grants, they're talking to a million people at once. It can sometimes be hard to filter through all of these emails.
Like if somebody sent me an email right now that was like, hey, I saw your poster, can I ask you questions? I would die of excitement.
Yeah,
Like that's like what we're doing now. That would be so cool to get, like, you know, outside of the people that I work with, to have someone reach out to ask me about my science. That's awesome. So don't be scared to that, because like that's such a cool experience.
Especially for the presenter who's presenting. And if it was some other question, like if you were interested in Matt's lab and you wanted to know more how to donate or how to get involved with the research he's doing, that might be a better instance where you'd want to reach out to the principal investigator. So the person who is running the lab. Go online and find, oh, all of these are number 1. So at the same time, you do all of these steps. Number 1, again, find a professional email address online. Oftentimes on posters, usually not abstracts, on posters, it will sometimes have people's professional email addresses, but look for something, it's often a .edu, or it has the name of the hospital or institute that they're working in, so a professional email address. Because you might be able to find people's Gmails online, or their LinkedIn email address that's connected to that. Probably not the best idea. And so once you find their professional email address, drafting up an email. And I'm always worried when I draft up an email, or what we call cold email, emailing someone that you don't previously have a relationship with. I'm worried it's going to go to spam, or they're going to see it and be like, I don't need this, and delete it. So something like this conference follow-up, or this conference question. I've had, after conferences, this conference follow-up, I click that email first. I'm so excited to see what someone wanted to ask me post-conference. Introduce or reintroduce yourself. Again, that you're a patient advocate and you're interested in this because, and then identify your question for reaching out. Like, this is just general, you guys know how to write an email, but to let them know that you are hoping for a response back, because you could send an email and they'll be like, oh, this person loved my stuff, thanks. And they don't need to reply because they just think it's a one-sided interaction. But you can say something like, looking forward to hearing from you, like if you had a question, or thanks in advance for any information you can provide, any clarity you can provide. Some closing remark to be like, hey, scientist, I want to actually talk to you about this. And then you can go from there. And one of the weirdest things that I never knew how to navigate was waiting for a follow-up, or like waiting to send a follow-up email. Oftentimes people are really busy. You get back from conferences. Sometimes people take time off after conferences because they're already wrapped up their experiments so they could leave for a few days. So allow a week or two before you send another email, say, like, replying to that email saying, hi, I was hoping to hear back from you, just wondering if this email got lost, things like that. But I never knew what was the right time frame to do that in, and it was really awkward. So I've found now, after being in grad school for over 4 years, that a week or two is good, especially if you're reaching out to a principal investigator. They can be really busy right after coming back from conferences. So yeah, give it a week or two.
And they probably just forgot. Like a lot of again, everybody's excited for research. They want to talk about it. So if you don't hear back, it's not that they were like, I don't need to answer this. It's probably just like, oh, okay, I'm going to put that on my to-do list of 8,000 other things to do. And they just got distracted, whatever. So it's totally okay to send a follow-up email.
I will say academics like filters are super aggressive.
Yeah, that's very true.
So sometimes it is like if you're sending from Gmail, it's usually okay, but like Hotmail, Yahoo, and everything, like older clients at this point usually go to spam. So
Automatically, we do nothing about that. And so sending a second email as a reply can sometimes bump things up from our spam folder into our inbox. Or if you're not getting a response, you can always try like one more time sending it from a different email address. But yeah, like the CU Anschutz filters are strict. So I've lost emails before that didn't come from another .edu account, but yeah, Gmail is usually pretty okay.
And worst case, you can usually find the guy's phone number and leave voicemail, and then you're like, oh crap, you know, I can talk.
Yeah, that's true. I know most PIs do not answer their lab phones, but that might just be specific to mine. Voicemail, that was a good point. They're not gonna answer, it's a voicemail, yeah. Sure.
I don't know if it's really a question, but so this particular conference is a little bit unique in that there's so much emphasis on patient advocate attending, right? And the scientists that are attending know that there's patient advocates, right? So, and I wanted to just kind of contrast this with, I went to another research conference and there were patient, when I was a patient advocate, and I don't know if some of the scientists knew, like, I even said, like, I'm a patient advocate, there's, are you, like, we're scaring you out?
Such a good point.
And so I think, you know, making sure, you know, I'm guessing that most people at this conference, if you say I'm a patient advocate, or if you get an email, but it might even be helpful in other situations to say, like, have you worked with a patient advocate before, or have you talked to a patient advocate before? Because I actually ran into a really interesting, I'm not gonna go into it, but an interesting but early career folks, and I was just talking about patient advocate, and they just like had no idea really what I was talking about. But I think that's right, like,
Yes,
yeah.
no, that's a really good point. I mean, I think our group is also unique, like acutely aware, but you're right, a lot of trainees will have zero idea what a patient advocate is, or even a patient care about basic research. You're right, that's not.
And in this particular case, like I'm. So like when these young career folks, that grads and postdocs, when they realized that I wasn't like a researcher, that they might get a position with something, they just kind of blew me off, and I was kind of like, well, you're lost, right? Like you totally could have gained something from this, but I think other people may not be in that kind of position to take that kind of response. But I don't want to also make it seem like all scientists are that way, but I think it's just something to be aware of.
I mean, the truth of the matter is, for me, like being so new to this, I'm not sure the difference between a patient and a patient advocate is just because I'm advocating for myself that it makes me a patient advocate.
You are here, you are a patient advocate.
You're not there as a researcher, you're there as a patient, that makes you a patient advocate, yes.
Okay.
I didn't know the definition either. In my field, we don't really have patient advocates. People get bacterial diseases and they either get treated with antibiotics or something worse happens. So, not really, that's what I remember with this. So, that's a great answer too, because, any of us could find ourselves in a position where we become a patient advocate one day. Good to know. Maybe do you want to take the takeaways?
Sure, I'll do the takeaways. So just as a reminder, the poster sessions were initially made to help disseminate the research and spark conversation between scientists. We're very glad though that we are starting to get patient advocates coming to these conferences. Researchers really want to talk about the research. There's a reason that we are dedicating our lives to doing this research, because we care about it, we're excited about it, and we want to talk to people about it. So yeah, just keep that in mind. We might be nervous about it, but we're still there to talk about the science.
We're not doing the research because we're getting paid a lot.
I love what I do. I love what I do. If there's an abstract booklet, use it to help prepare beforehand. So that way you're not inundated. Like conferences are busy. You want to get from this speaker, that talk, you want to get coffee here, all of this, it gets really complicated and busy. So if you have access to that abstract booklet, looking at it ahead of time, even just like the day before, going through and highlighting ones can be really helpful in getting you to the posters that you can get the most information out of. If you're looking at a poster, either in person or virtually, look at that title, look at the abstract, and that conclusion to help get those keywords and important points across to help you decide if you want to engage further with that poster and that researcher. And then just like all the excellent points Laurie was talking about with active listening, they want to talk, but hopefully you want to be there to listen and to have a conversation. So just be mutually respectful when you're at these poster sessions. I think that was pretty much all we had.
Here's our
Here's our question slide, like Laurie said you're gonna.
But you guys are fantastic about asking your questions and providing your comments throughout. So if anyone has anything generally you'd still like to ask.
Just a quick comment. LBCA does summarize all the ILC related posters from SABCS into lay summaries after the conference every year. So I don't know how many, I'd say there's probably 20 ILC posters maybe at SABCS. So right after the conference, 3 or 4 of us at LBCA that have scientific background will write lay summaries of all the ILC research that was presented. And I think you can still look up last year's summaries on LBCA website if you're curious, just to kind of see what those look like and give us feedback like, hey, I still really had no idea what the heck that was about. More lay, that's too simple, more science, but they are there for you to use at your leisure, and we'll do that again this year after the CBCS.
Yeah, if you ever have any comments, questions, concerns at any point from here until forever, you can email us there or ask Matt and he can get you in contact with us.
All right. Let's talk some biology. Okay, so this is actually kind of a new thing for me as far as the crowd I'm teaching to today. So usually I teach graduate medical students cancer biology. Usually I have like 2 to 3 hours to do so, and usually I'm on like a hyper-specific topic. But I'm going to attempt to cover cancer biology in a reasonable amount of time this morning / afternoon. Lunch is going to show up in a little bit. Please get up, get lunch while we're doing this. I'm going to try to cover broadly what is cancer, what is progression, give you some context for normal cells focusing on the breast and how they become cancer. I'll talk a lot about breast cancer subtypes, since I know all of you have pathology reports that talk about subtypes, and that's at the forefront of treatment. I'll talk a little bit about how subtype informs biology and how it is biology. It really is, we target therapy more to the biology than is easy to appreciate from pathology reports. And of course, during the whole thing, I'll fit in lobular, and then at the end, I'll talk about kind of where paradigms are going and some of the major themes at the conference coming up. I will make these available. So most of the figures I have are from these two general reviews, which are kind of like broad disease primers for breast cancer as a whole. And I'll make these PDFs available to the group. They're written for scientists, but they do still cover like everything, at least in a broad stroke. So that's where I'm getting most of the figures. Please stop me. If the last discussion was fabulous. I want this to be interactive. The stuff I'm going to put up is going to look daunting, but we're going to chew through it together. All right. Before I get into breast cancer, I wanted to spend a minute on the central dogma, because you're going to hear some of these words over and over and over, and I want it to be as clear as possible when I say these things. So the central dogma in molecular biology is that all of our cells contain DNA, and that is essentially the instructions for what that cell is. It's the same in all your cells, and how it gets read out is what dictates the structure of that cell, essentially. DNA gets read out as instructions into this molecule mRNA. There's a lot of other things that happen, but mRNA is what we're going to focus on first. This essentially represents how this cell is reading off the instructions that are unique to that cell. So the information goes from DNA to RNA. That RNA leaves the nucleus where the cell lives. It gets read by machinery called ribosomes and is turned into a protein, which is also called a polypeptide. That is typically what actually executes the change in biology in that cell. Typically, we have an RNA biologist in the room, so I want to be careful with that. But so that the dogma is information goes from DNA to RNA to protein. When you think about genes in a cell, those are pieces of information that live in the DNA, and those are the functional units that get read into mRNA that then become a protein that makes cell biology happen. Okay.
What do you mean when you say they get read into RNA? Is that what you're saying?
Yeah. So this is all of the instructions to identify, like, we want to execute this program. The cell has to take some of this information and turn it into this kind of, it's called messenger RNA because it is the message that the DNA is sending out to become messenger RNA. So that's what the M is here.
Oh.
Yeah. It's also like, it's from a practical perspective, all of the instructions, it's 3 billion letters in your genome that is wound up basically in a ball in the nucleus. And to get it to the machinery that makes it protein, you have to have an intermediate. And that's what this represents. Please.
So bad.
Please.
David talks about this all the time in our presentations.
Please.
So I like to, so I have a sweet tooth, so I like to explain this in terms of cookies. So I like to think of your DNA as a cookie cookbook. So it is a recipe book that has all of the recipes for all of the cookies you ever want to make ever. But you don't want to name all of those cookies all of the time, right? So RNA, I like to think of as a photocopy of a single recipe in that book. So you want chocolate chip cookies. That recipe book has the recipe for chocolate chip cookies. You make a photocopy of that, and you have this temporary copy of that recipe that you can then read from to make cookies, which in this analogy are the proteins. So this RNA is temporary. The cell can decide how many photocopies you want. That also helps determine how much or how little of the protein or cookies that you're going to make. So yeah, the RNA is this temporary copy of a single gene in your DNA. If that helps.
I'm going to steal that.
I always think of it in terms of like a Xerox copy now.
I like doing that because I usually get to eat cookies while I present.
I think there might be a couple left. Okay.
I have one more too that I feel like it's important to understand. Because kind of how like you can take the recipe and make the cookies can't go back into the recipe. So this is only a one-way street. And I feel like that's also really important to understand too.
There's a few niche things that break the rule, but
Yeah.
they're super niche. This holds. Yeah. All right. There are some terms we use a lot that also overlap in this dogma, and I want to attempt to clarify them because you're going to hear, because I'm trying to put up examples of very common terminology here. Okay. If someone says, or you hear in a commercial or whatever, I have the breast cancer gene. This does not mean that the cancer cells or the patient have a gene or unit in their DNA that is unique per se. What it almost always means is that the patient has an inherited mutation or I don't want to say defect, but it's a variation in that gene, a change in the underlying sequence or code, the letters that are in the DNA, this level of information, which in the case of breast cancer are genes that are associated with breast cancer like BRCA1, BRCA2. Those are the two we hear about the most. The mutation is a change in the letter, in the code in the DNA that then carries over into the mRNA. When that gets photocopied, thank you, that is in the photocopy now. And that's what's something unique in that photocopy. And then when you do the recipe, the cookies don't come out as well. Man, I'm going to use this now. And then there is some change in the biology that imparts cancer risk, and that's another lecture. But when you hear somebody say, I have a breast cancer gene, it's not that they have a gene that's different, it's that there's some variant that causes a defect in the cookie, and that imparts cancer risk. The terminology does carry over. So what you might hear at a conference, oh, my pointer's, I keep forgetting it's over there. You'll hear somebody say, like for Lobular, that the E-cadherin gene was knocked out in the cells in the lab. We use that term a lot, knockout. What this might mean is very different than the breast cancer gene use, but that perhaps various lab methods were used to either delete, like literally remove the gene, or to catastrophically mutate it, just to put as many errors in it that it just wouldn't work. We did that in the lab. It's something very different than what would happen in a patient. We can then, that carries over, we render the mRNA absent entirely, or it's otherwise defective, and then we get a dysfunctional protein. So we use that in that case, that's another use of the word gene in a laboratory setting.
So you ripped the page out of the cookie.
Oh man, yes, yes,
And you're making it from memory.
yes. You're ripping it out. The word gene here does get used at two levels. So we've talked about how it gets used at this level of information. It also gets used here. And when we, as scientists, when we say it, we use context clues to know what it is, but that doesn't help any of you. What you'll hear are things like levels of the gene were increased in these tumors, or expression of the gene was decreased. And that is referring to the mRNA, the readout or the number of photocopies. If we're saying levels of the gene went up, it usually means more photocopies, more amount of that. Or if expression is the word we use a lot, that means you have fewer photocopies. This is a major way that the cell tightly controls the number of cookies that are made, the amount of protein that comes out, is by controlling levels of the gene. So your levels, expression of the gene. The use of gene there refers to mRNA, whereas in other settings it refers to DNA, just to make it harder on all of you in understanding what we do. Finally, similar words that you'll hear, the tumor expresses the ER protein, it's ER positive, ER negative. We might say protein levels were decreased. Sometimes expression gets used interchangeably for proteins and genes, again, just to make it harder for all of you. That's referring to the end product of the dogma, which again is typically what is affecting change in the cell. It's the cookies that came out of our analogy. So I'm going to say things, and you are going to hear things like this in conferences, because this is the terminology we use to describe our science. But hopefully this is at least a primer to help you dissect that. I really like the cookie analogy. All right. This is the only slide I'm going to read to you, and this is just so we're all on the same page of terminology, because you've undoubtedly all heard these words. So cancer, by like the biomedical definition, is an abnormal growth of cells caused by genetic changes, which I will call hits a lot for the next chunk of time here. These hits or changes to the genetics, which can be mutations, deletions, removing the copy, things like that, lead to dysregulated proliferation or growth. It changes how the cells multiply and divide. This leads to the evolution of a population of cells that can eventually invade local tissues. The property of invasion is called malignancy, and then ultimately distantly metastasize and lead to the death of the host. That, in a nutshell, is what we say when we use the word cancer. The word tumor is actually a non-specific term. It can mean a lump or swelling, and it doesn't necessarily link to cancer. So you may hear tumors in other settings, but it is an overly broad term. And not all cancers form tumors. Any leukemias or other liquid tumors never form or liquid cancers never form a tumor. Neoplasia is also a word that's kind of used interchangeably with cancer, and it encompasses conditions of abnormal cell proliferation, which is called hyperplasia. That's often the precursor stage for cancer, and I'll talk about that in a minute. It can also mean changes to the tissue architecture or how cells form in their particular organ. That's called dysplasia. And neoplasia then can be benign, which means it cannot invade beyond its local context, or it can be malignant, and then it's locally or distally invasive. When a neoplasia is malignant, it is cancer. So that's where all the terms fall in. Okay. I'm going to treat you all like medical students for a little bit. And in that context, they also come in very different backgrounds, but this is how I explain it to medical students too. So please stop me if anything doesn't make sense. This figure here is from kind of one of the more famous series of papers in cancer biology called The Hallmarks of Cancer. And one of the things that we've tried to do as scientists over the last long time, 50, 60 plus years, is understand what changes in a normal cell to make it become a cancer, to do all of those things like locally invade and to get around all the things that our body does to stop cancer from happening. And this is just like a snippet of all the things that a cancer cell has to be able to do to survive as a cancer. It has to keep growing nonstop. It has to evade the things that would stop it from growing. It has to evade the immune system. It has to effectively become immortal, which our cells are not normally immortal, and so on and so forth. There's a lot of things that have to go wrong for a cell to become a cancer. And how they go wrong are these so-called genetic hits. I'm using this term broadly to describe anything that happens to the genome, to those instructions to change either the sequence, the code, the actual recipe, or change how the recipe is read out. All of those changes represent hits. The hits can come from a whole bunch of different sources. That includes ionizing radiation, like X-rays are related, UV radiation, obviously those are directly causative in things like melanoma, that's the literal hit. Industrial pollutants, cigarettes cause direct damage to DNA that cause those hits. Exposure to metals or other toxins can cause that, and sometimes that's changes to the DNA, sometimes that's changes to how the recipe gets read. Viruses and bacteria can cause cancer by related mechanisms, some again directly on the DNA, some by breaking the messages. The fact that we breathe oxygen is problematic because it does cause damage, and that's just part of our aging process as creatures that are aerobic and breathe oxygen. We alluded to earlier that 5 to 10% of breast cancers are related to hereditary mutations. So you already have essentially one break, one hit in a gene, and that changes essentially the math for how cancer happens. And then every single cell in your body has 6 billion letters in it. And it's amazing that they get replicated the same exact way every single time, as often as it does. Every once in a while, there's an error. And more often than not, the errors cause nothing. Nothing happens, but occasionally you get a bad one, and it's a tantamount to bad luck. All of these things are just different ways that can cause mutations, deletions, different defects in the DNA itself, epigenetic changes, which is changes to the structure, like how the DNA is folded on itself and wrapped up and contained in the nucleus. All of these things represent hits, that I'm going to just use that word broadly to describe all of those things, because all of those things can change either the instructions themselves or how they get read. And that's kind of what we refer to in that space. I want to give you a little context for how those hits then translate to cancer. So cancer incidence, I'm sure you're all familiar with in principle, increases in incidence with age, with the exception of some pediatric cancers because the genetics are different. Most solid cancers occur more in adults. In men, prostate is the most frequent and increases the incidence in 40s and 50s and into 60s. Same with women, breast is most common, but all of these solid tumors occur more later in life. The numbers get funny when you get this far out, which is why the curves dip down again. But in general, cancer incidence increases with age. And the main reason for this is that it takes a lifetime, literally, to acquire all of the different hits required for a cell to become cancer. That wheel I showed you a moment ago of all those properties, it takes a lot for a cell to gain all those characteristics and evade all of our body's defenses otherwise. So it's kind of a mathematical marvel that this happens at all. With that, though, not only is it lifetime, but cancer requires the accumulation of these hits over decades. And there's some really cool epidemiological data to support that. So on this graph, on the left axis here which is the red line, is cigarettes smoked per year in the billions. And then lung cancer incidence is in the green line, and overall non-tobacco cancers are down here. Smoking went up around World War I, the Great Depression. There were a couple of dips, for war and stuff. But then it took 30 to 40 years for lung cancer rates to went up. Everybody was smoking a lot during that time, but it didn't cause cancer right away, because even though these patients were exposed directly to a toxin that was causing hits in their DNA, it still took 30 to 40 years for those lung cells to acquire enough hits to become a cancer. So our bodies are actually really good at fighting it. And there's a lot of reasons that things go wrong, but it does take time, and that's why it's typically associated with age. Another cool one is mesothelioma risk in insulation installers, and this has to do with asbestos exposure. So this is cumulative incidence of mesothelioma and the age at which the patients were first exposed. So when did they start as installers? And it didn't matter how old they were when they started installing, it all has to do with the time since they started installing. So it's not just you get cancer because you're older, it's because it's related to cumulative exposure. So it's the same exact amount of time of asbestos exposure to cause the cancer, regardless of how old the installer was when they started. Again, not driven by age per se, but by cumulative exposure. And I can't stress enough that so many of these hits are not things that anyone does. It's just living as a human. It just those are where the hits come from. So as I mentioned, one cell has to get all of these hallmarks, and each of them essentially requires one or more hit. Sometimes a hit can cause more than one of these things to happen, but all of these things represent different control mechanisms, different genes that have to get hit or broken to allow the cell to do these different things. So if you have a population of cells, each of these gray dots is a cell, let's just say in the breast, one acquires a hit, and it's a hit in a gene that's important for how the cell grows. That cell, the mathematical likelihood, aside from cigarette smoking and direct exposures, the likelihood of any one cell getting a hit is roughly 1 in a million. So to have enough cells just to have the mathematical likelihood of a second hit, this one cell has to expand to something like 1 million cells. Our cells in our body don't typically divide that fast unless you're in kind of wound healing or other specific tissues that do that. So it takes a long time. It takes years for that cell that has one hit to expand to become a whole bunch of cells. And then one of them will acquire a second hit by mathematical chains. That one has to expand, so on, so forth. So for every million cells, 1 10 to the 6th or 1 million is about equal to 2 to the 20th power. That's relevant because every time a cell divides, it doubles, right? So it takes roughly 20 cell divisions on a population level to get to 1 million cells. That takes a long time in a normal healthy tissue. So between the relatively low likelihood of those hits and the fact of how long it actually takes, acquisition of hallmarks takes hand-wavingly 10 to 15 years, and then the entire succession takes 40 to 60 years, which is why, again, cancer occurs later, because it's just due to cumulative exposure and collection of those hits. Yeah.
Take a step back. So does the circle there and the words underneath it mean that in order for cancer to develop, it has to have all of those things?
Basically, there are intermediate steps, and that's
ultimately it's not like you can have half of them.
Not really, yeah.
Okay.
Yeah, so I guess just to poke at those for a second. So sustained proliferative signaling. This means the cell has lost its normal context, that it stops growing. Like your skin doesn't just keep growing. It grows when it needs to. If you get a wound, there's an initiation of cell growth to fill the gap, and then it stops. Those on-off signals are really important in all the tissues in our body. Cancer has to lose the off signal, which is evading growth suppressors and sustaining proliferative signal. It has to figure out ways to lock on an on signal and ignore the off signals. And that takes a lot. Deregulating cellular energetics. Your cells all eat sugar, amino acids, fats. In order to make more cells, they have to consume more fuel. And making a cell so it sucks in more fuel is also more series of hits that it sucks in more of, and the right kind of fuel it needs to make more cells. Resisting cell death. Our cells are really good at killing themselves when they recognize that cancer is happening. Our cells actually have cancer protective mechanisms that go, oh, this cell's growing and it's not supposed to, kill it, we'll just make a new one. So a cell has to turn off a whole bunch of processes. The one that maybe you've heard of as P53, which is mutated in a ton of cancers. That's like our chief defense. And that has to get broken more often than not to allow the cell to do any of this. So genome instability, this one's a cool one, biologically cool from a tumor. So BRCA1 and BRCA2, their loss doesn't cause cancer per se, but it increases the rate at which a cell acquires hits. So you just supercharge the rate of the whole process. There are actually cases where, ovarian cancer in particular, the cell will they'll turn off the genome instability to resist chemotherapy because they're done with it. They've already become a cancer and they don't need to be unstable anymore. Cancer is a jerk. Angiogenesis is one that happens later. So this is actually a cell co-opting the vasculature to bring in a blood supply. And a cancer doesn't have to do this until it wants to get big. Because for a while, it'll just feed off whatever local vasculature is nearby. But as it grows, it gets literally too big to diffuse food and oxygen exchange from the vasculature. So it grows new veins. Yes?
This lobular like that, the way that it spreads.
That is a great question. So does lobular grow vasculature the same way? Shrug. Yeah. Yeah. Yes and no. It definitely needs to do different things because it doesn't have the same mass. It does still, even though ductal forms a solid lump, so it has no exposure to lymph vessels or blood vessels, so it has to. Lobular is different in that we think the way it can co-opt vasculature is different. Probably not a satisfying answer, but yeah, a little bit. But so it's not, and the question was a moment ago, it's not an all or nothing approach. As the cell acquires hits and you get masses of cells that all share those hits because they've divided and expanded, cancer does progress down a stepwise progression. It just happens that in some cancers we know more about it or less about it because we can see it. So obviously colorectal cancer, you can put a camera in there and look at it so you can see. Breast is fairly straightforward to biopsy, so we can see. Things like pancreas, we have way less idea because we can't ever get a look at it. But in general, what this happens is you have some normal tissue, you'll have a cell that got a hit, it will eventually expand, and and I'll show you more images of this directly. You'll have as an expansion of cells that look normal-ish, but only ish, and then eventually they'll look less normal and less normal. They'll look very much not normal, but they'll still be contained, and then eventually they'll invade, and that's when cancer happens. So these early stages where the cells are still normal-ish are termed hyperplasia or dysplasia, and the equivalent in breast is usually atypical ductal hyperplasia or atypical lobular hyperplasia, where you have maybe more cells in a space than there ought to be, but they still look normal, and I'll show you in a second. Later stage is called carcinoma in situ. There's a lot of argument about removing the word carcinoma from this diagnosis because it's confusing. And in this case, though, the cells are not normal, but they are still physically bounded entirely by the normal structures. So they're not invasive yet. You'll have all these slides too. And then eventually that becomes an invasive carcinoma. All of these early stages before invasion are what's a benign diagnosis. So if that's in your pathology, including in situ, those are benign. But once they can leave their normal structures, that's when you have malignant, and that's cancer. Again, I'll show you pictures of this, but in this progression, as I alluded to, it's not an all or nothing, and that as cells are moving down this process, we know they're acquiring more and more hits. And there have been genetic studies in a lot of cancers, colorectal is kind of the poster child for this, that we can track that as they move down this progression, they have more and more genetic mutations, genetic hits. So it is a progress. In the breast, it is very much a multi-step process. I mean, we've heard of these atypical and hyperplasias and in situ lesions. So this is a cartoon then of the normal breast structure. In general, you have these milk-producing lobules that are connected by ducts, and that's what moves the milk from the lobules to the nipple during lactation. In any one of these, if we think about the lobules, these are the terminal ductal lobule units, and this is the milk-producing part of the ductal lobular tree. And then this is a cross-section then of the duct. I will start here that the terms ductal versus lobular are a very unfortunate historical artifact that don't actually mean anything that we know biologically. When lobular was first identified by pathologists in the 50s, they happened to see it in the lobule. And we don't actually have any hard biology evidence that those two things are related. So it is a super irritating historical artifact, and I've even yelled at Komen and BCRF on Twitter when they tweet these things. There might be biology, but we don't actually know that yet. So I'm going to keep saying ductal and lobular, ducts and lobules, but it's not connected, and it's really irritating. So if we think about this cross section of the duct, though, you have a whole bunch of supporting cells, like the normal breast structure, fat tissue, immune cells, other structural components. There are these myoepithelial cells, which are what can contract the duct to move milk along. And then you have these luminal cells on the inside. And those are typically, not both of them can go wrong, but those are what we're thinking about typically in terms of lobular cancer. It's those interior cells on the inside of the duct. But during the process of progression, if we're thinking about that cross-section of the duct again, early hyperplasias will look like just more cells crammed into that ductal lining. They'll be like squished, and there'll be a few more than there ought to be. In carcinoma in situ, they can go so far as to fill the duct, but they're still bound by the myoepithelial cells, and there's a membrane around there that keeps it all together. In situ lesions are still wholly physically constrained by that. As soon as they leave that structure, though, it becomes an invasive carcinoma. And that's when it's cancer, and that's when it can spread. So again, in terms of the breast, we do know that this is a similar process. This is a cross-section on histology of normal breast ducts. You can imagine there's a mixture of a whole set of ducts that are running through the breast, but also some of these will represent those lobules. All we have is a cross-section, so unless we have depth, we can't really tell what's what necessarily, but that's what we're looking at. This is zoomed in, but this is an atypical hyperplasia. We're like way zoomed in on a duct here, and the duct is also very distended from its normal size. Didn't project great, but the boundary, the normal, that luminal layer is kind of out here. And you can see, obviously, there are groups of cells that are growing out from that into the duct. It hasn't, like, it still looks like a duct. You still have these kind of, the boundaries here, if you could see them better in the projection, still look kind of like it's supposed to. So that's why we're not cancer yet. It's just a hyperplasia because we have way more cells than we're supposed to have in the duct. This is an example of a ductal carcinoma in situ. It has basically filled the duct. None of these cells look like a breast duct cell. They have completely forgotten who they are, but they're still bounded, and you can kind of see that membrane on the outside and that layer of myoepithelial cells there. So it's still bounded, so it's in situ. But then as soon as it leaves, in this case, this is a ductal cancer. It looks nothing like a breast. It's just a whole bunch of cells that have, you know, run rampant through the tissue. So that's an invasive cancer. Lobular carcinoma does, in principle, the same thing, and I'll come back to your question in a second. But these are examples where we actually see lobular carcinoma in situ adjacent to invasive cancer, and you can actually see where it has left the normal structures. So these are all 4 examples from different tissues, actually, of the Pittsburgh group, where they identified an in situ lesion and identified some cells that had been able to invade. So those cells are what acquired the hits to break through that local membrane and become invasive. We have, as I alluded to with the one question, we have a really bad handle on figuring out which of these in situ lesions are likely to have the hits to get to invasive cancer. It's not clear whether it's a question of inevitability, because it's really not. Many more patients will die with an in situ lesion than of the invasive cancer. Like the corollary in men is that almost all men will die with prostate cancer. Like if you cut open a 90-year-old man, they have prostate cancer, but it was likely never symptomatic and never outgrew. The same holds in breast. We think a lot more women have these in situ lesions, and that was one issue with mammography, is that mammography is really, really good at picking up in situ lesions, but we don't know necessarily risk for progression. It's a lot of guessing, clinical markers, blah, 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 blah. And that's why women with in situ lesions do typically get aggressively treated, because if they have cells with this many hits that they've forgotten who they are, it's just likely that, you know, than any random patient, that they're going to get an invasive cancer. The lobular versus ductal in situ thing is really weird, and I don't know why it's really weird. I haven't gotten satisfactory answers myself. Ductal carcinoma in situ is considered an obligate precursor lesion, meaning like this in situ lesion has the potential to develop into an invasive cancer. I really don't know the reason for this. Lobular carcinoma in situ is not considered an obligate precursor, meaning clinicians don't look at that and go, that might become ILC. They look at that and go, there are cells in the breast that have mutations, so that there's an increased risk. So that distinction changes the aggressiveness of the treatment. So that's our introduction to genetic hits and progression from normal tissue to cancer. What I'm going to go on now is that This is obviously stylized and generic. I'm kind of showing you, two types of breast cells, and it becomes a quote unquote cancer. Obviously, you're all here because you're interested in lobular cancer, so there's already multiple different types of cancer. Within those types, there's histological subtypes, which is what it looks like, and intrinsic or clinical subtypes, which has to do with the biology. We've heard a lot of these words, and I'll go into this. Before I go there, any questions about hits, the cascade, the process? Any words I've used too much that I don't realize I'm not being clear? All right. Okay, so let's go back to that diagram of the different types of cells in the breast. The functionality of those luminal cells versus the myoepithelial cells is very different. The myoepithelial cells are contractile and they can squeeze the duct to move milk along. The luminal cells are in part would have to make milk and maintain the environment in that ductal space. So their function is very different. So they have to behave very differently. The way they read out to their DNA to the recipes is very different. So you can imagine then that if either of those cells start acquiring hits, the biology of the tumor downstream is plausibly going to be very different. So when we think about the development of the normal breast, in general, we're thinking about some sort of stem or progenitor cell. And this is a cell that is a relatively blank slate. There are different types of stem cells, whether they're embryonic, whether they're adult, what tissue they reside in, but there are cells that are effectively a blank slate in a given tissue and can be anything that the tissue needs it to be. That stem cell will divide for various reasons under various environmental cues to become what are termed a basal progenitor or a luminal progenitor. The basal progenitors are thought to then go on and become the myoepithelial cells that are contractile and can line the duct. The luminal progenitors are what then go on to become the inside of the cell, and there are different flavors of those as well. Some of them are hormone responsive, some of them are not. And they all come in different, we're starting to get we think a handle on kind of different steps along that cascade because it's not a one-time process either. What is diagrammed here is that if any one of these different levels of what's called differentiated cells, when they kind of decide who they are more and more, if those acquire hits, it's likely to lead to different types of cancer. So the basal-like or triple-negative cancers are thought to typically come from actually luminal progenitors, but very early ones that have only just started to decide who they are. If we get hits in more mature luminal cells, things that have gotten closer to being, I am a hormone responsive luminal cell, those are what likely become estrogen receptor positive cancers. If you get a mutation way up in that stem cell, those early progenitor cells, those become a particularly aggressive type of triple negative breast cancer. So what I think is fascinating from a biology standpoint is that if we look at gene expression, the levels of those different recipes getting read out in different types of breast cancer, we see evidence that they still at some way or another remember who they were. And we can see evidence of that in gene expression. So these are examples of luminal genes, or these are genes that we know that are turned on more specifically in those mature luminal cells in the breast that aren't typically turned on in the other cells. That includes estrogen receptor and progesterone receptor, which we'll talk more about. But if you look at tumors that are clinically termed, and I'll get more into the subtype names in a moment, luminal A or luminal B, they look like those luminal cells from the normal breast, whereas those triple negative tumors don't express any of those markers that we kind of refer to them as markers of cell identity. Like the cells still remember who they were. Same with basal genes. So these are genes that are expressed more in those myoepithelial cells. Blue is low and yellow is high relatively. So those luminal tumors don't tend to express those basal markers, whereas the basal ones do. Again, those are markers of who the cells should have been or who they were before they went down this tumor genesis cascade. And then there's markers, these are keratins, these are kind of intracellular. They live inside the cell and provide structure. We even have evidence of those being high differently in either luminal or myoepithelial cells, and the tumors reflect that. Point being, that's this kind of idea of cell identity in the breast is, we think, a lot of what dictates what type of cancer happens, at least in this case, at the level of estrogen receptor, progesterone receptor, etc. The histological subtypes are a layer on top of this. And what we think is what's happening is that the specific hits that any one of those different cascade of cells gets, if they are different, they will drive different types of breast cancer. So ductal cancer is not actually a definition, it's a default. If there is no special histological diagnosis, it's ductal. There's a movement to actually call it not otherwise specified or no special type. These are examples of the special types of breast cancer, and lobular is the most common amongst those at 10 to 15% of all diagnoses. For all of these, though, don't worry too much about the different types, but the idea is that we already have hints that the specific hits that happen are different, and that changes the cell architecture, it changes their behavior, so on and so forth. And we'll talk more about it, but chief amongst them in lobular cancer is loss of the gene, loss, mutation, deletion, etc., of the gene CDH1, which encodes the protein E-cadherin. And we think in the community that it's loss of that protein, it's that hit or mutation that, long story short, causes the lobular phenotype. And you can see that in these blue, there's a bunch of other mutations that are specified for these different histological subtypes. A lot of them are super rare in breast cancer overall, except for in those subtypes, which is one of the reasons that we think that those specific hits drive the biology.
Those capitalized things are proteins?
Genes, yeah.
Genes.
Yeah. To make it again harder for everyone, we often name genes and proteins differently. If it's if it's all caps and italics, we're referring to a gene, a human gene. The protein name is not italicized and may be different. So the gene CDH1 is formally called cadherin-1. It encodes the protein that we call E-cadherin. Those are all the same thing effectively.
So that would be the gene that got the hit.
Yes.
Okay.
Yep. That is the gene that got deleted or mutated in lobular cancer. All of these rare tumors have mutations in genes, like in this case, MIB. This fusion, it's a fusion of two different genes in the genome. These things don't happen in other settings. So it just gives us a lot of confidence that whatever this is because of this. And that's the same in lobular. But E-cadherin loss and deletion is just way more common, and we don't know why. Okay, so we've talked about histological subtypes, which is our favorite and why we're here. That then breaks out into those intrinsic or clinical subtypes. This is in one of those papers I referenced. Again, I'll get everybody the PDFs so you can see this yourselves too. But we're thinking about pre-invasive or those in situ lesions versus invasive disease. We're thinking about ductal histology, which again is kind of the absence of fancy stuff, versus lobular histology and other uncommon subtypes. And then that each of those can break down at different frequencies into each of these clinical, or you'll hear clinical subtypes, you'll hear intrinsic subtypes, you'll hear PAM50, you'll hear IHC4. Anything to do with subtype is typically relating to these. And the way those break down, where did my mouse go? There it is. Luminal A and luminal B are the ones that are relevant in lobular cancer most of the time. There are, of course, other situations. These are basically all tumors, and I'll go into these biomarkers more in a moment, that are essentially all ER positive. They may or may not be PR positive. They are typically not HER2 positive. Again, I'll go into this more. And really the big difference is proliferation. And we have kind of like, we have the more refined measures of luminal A versus luminal B if we do gene expression profiling and like more in-depth tumor profiling. But the like hand-wavy clinical way to decide this is level of Ki67, how proliferative the tumor is. And those are then kind of tied to risk of recurrence-ish. Yes?
So if your patent report doesn't use the word luminal at all,
It probably won't.
then if you're PR positive, HER2 negative, with a high Ki67, are you?
You'd be clinically considered luminal B. Yeah, so these subtypes were defined based on gene expression, where they took out a chunk of the tumor, they popped it open, they read all the mRNA and got levels of every single gene in the genome, and then used that to distinguish the tumors. We don't do that. We get IHC for ER, PR, HER2, and Ki67. So they've basically taken those four and distilled them down into the same subtypes, which is they're used interchangeably. Whether that's fair, the jury's out. And I will also reinforce that all of those subtypes, that gene expression subtypes, was all based on ductal. The initial study that was done back in 2000, published in 2000, was 70 tumors. 5 of them are lobular, and 3 of them were like super weird niche lobulars. And I don't know why they were in the study. It was like an accident. So I will also hand wave that we as a field are trying to figure out how much those subtypes matter for lobular. My lab's one of the things we're trying to do in the MetaViber grant that Donna's on is get some of that data just for lobular and say, what are the actual subtypes? Do we see what's the big tumor profiling studies nationally have seen? Yes. I sighed because the jury's out. For ductal, 14% is the cutoff. That's clinically what's used. There's data for lobular that 14% is not a relevant number, that it's more like 5 to 8. But again, the jury's out. And I don't want to tell you that your tumor is high risk or not because of that number. Point being, it's weird in lobular. And I hope we can do better than Ki-67. I don't know if that's a satisfactory answer at all.
What are the big guys on the lower? How are they different?
Oh, yeah, I'll come to that in a second. Thank you. Yeah, so the ones you'll hear about intrinsic PAM50 are usually these top ones. We talked about luminal A, luminal B. HER2 enriched is kind of a squishy subtype because the current thought in the field is that the gain of HER2, which is you actually get an amplification. The cells have more than 2 copies. They can have dozens of copies of the gene, so they get a lot more of the recipe coming out. That can happen on top of other subtypes. If it happens in a luminal, it falls somewhere between luminal B and HER2 enriched. If it happens in a basal or a triple negative, it becomes just a HER2 enriched. So it kind of is a squishy middle ground. But this is when you have that HER2 gene amplified, so if you're HER2 positive. And then the rest of them are flavors of basal-like or triple negative breast cancer typically. So your ER, PR, HER2 negative. I'll talk more about that in a minute. All of these subtypes is to inform treatment and risk assessment is what clinicians want this for. And that's kind of what it's being used for. Oh, thank you. That's the number of tumors that fall under that type. So overall, again, so if we think about luminal AB, HER2, triple negative, those are really how clinically they get divided, even though gene expression kind of teases them out further. It comes down to ER, PR, HER2, Ki-67. So these percentages match up here that the majority of newly diagnosed breast cancer overall, forgetting lobular, most of them are ER positive. 70+% of new cancers express ER. Whether or not they're low risk or high risk in here is based on whether they're PR positive or negative, or whether they're high or low Ki-67, and it's a continuum. We kind of call it high, low risk, but really it's a rainbow, a terrible, terrible rainbow. You can be HER2 positive and ER positive or negative. That's around 15% of tumors, and then around 50% of tumors are triple negative, meaning ER, PR, HER2 negative. Something that I tell anybody at the funding level who listens is that everybody wants to fund triple negative breast cancer research at 10 to 15% of diagnoses. Lobular is 10 to 15% of diagnoses, so it should get the same attention, but you all know that. These breakdowns are for breast cancer as a whole. For lobular cancer, 95% of tumors are ER positive.
So we talk a lot about the ER positivity,
Uh-huh.
But we also get what the PR is, progesterone as well. Does it matter if how high the progesterone percentage is?
Yes and no. Yes and no. I'll talk about it functionally in a second. Short version is that PR is overall considered a marker of whether ER is working. So in anti-estrogen, I could go off and this is my career. In endocrine resistance, many times the tumor will still be ER positive, but not really using the estrogen receptor anymore. ER won't be doing its job. It'll just be there. So ER is no longer a good marker of endocrine response or whether the tumor still depends on estrogen, right? PR is kind of a surrogate biomarker for, and it's not perfect by any stretch, but it's a reasonable surrogate biomarker of, is ER still doing normal ER things? And if that's the case, the tumor is more likely to still be dependent on the hormone estrogen and still likely to respond to anti-estrogens. That does not mean that PR negative tumors don't respond to anti-estrogen. That was just that's kind of the vague hand wavy. This is why.
But if they're both pretty high, then they're both going to be receptive to the estrogen.
We are far more confident that the tumor is likely dependent upon estrogen.
Right.
Yeah. Good question. Okay. Anyway, so this is part of why my lab focuses so much on estrogen receptor function, because it does encompass the vast majority of lobular cancers. It also ties into the fact that, and I could go on to this for another like hour, the associations between lobular cancer and estrogen are also very different than ductal cancer. Maybe like the chief one is that during the 80s and 90s, when women started getting more hormone replacement therapy during menopause to alleviate menopausal symptoms, the rates of all breast cancers went up. And that's why HRT had like fell out of favor. It's coming back for different reasons. But at the time, kind of the sneaky part was that it was only combined estrogen plus progestin therapy that increased the risk of ductal cancer. You can get estrogen alone, but if a woman has a uterus, unopposed estrogen will cause endometrial cancer. Progesterone blocks that. So that's why it's almost always combined. In women without a uterus, you can get estrogen alone and there's fewer side effects. Estrogen alone was sufficient to increase the risk of lobular cancer, but not ductal cancer. The takeaway biologically is that the way that those early lobular cells respond to estrogen is different. And that's been a whole thrust in my lab. And that's why part of, we don't know why yet, but that's part of why we think that this is a biological thing. And we're just trying to figure out why that's the case. I want to dive a little bit into those 4 biomarkers we talked about, plus ecadherin. This is not meant to terrify you, but this is like a snapshot of what we as biologists are thinking about when we're thinking about what's going on outside the cell. What kind of environmental cues is that cell responding to? How does that change biology inside the cell? What are all the pieces of what's called a signaling cascade that take outside signals and change biology? So what's happening from outside to get to inside? And then eventually, how is that changing how we're reading out the DNA? How is that changing how the recipe is getting made? Is it turning genes on or off? How is that changing the proteins that are getting made? How many cookies we are or not getting? So this is what we're thinking about when we have a HER2 amplification. How is that affecting the activity of all of these signaling molecules? How are we getting message from point A to point B? And how is that changing cell biology? So what I want to impart to you is that those 4 biomarkers are not just biomarkers. They are actually, they're relevant readouts of biology. I said we were going to 1 and we're going to go past 1. I'm sorry for a lot of reasons. Just noticed the clock. All right. Estrogen receptor and progesterone receptor. This is one I could like talk for the next 6 hours on. This is what I've been studying for like the last 20 years. These are molecules that typically live in and around the nucleus, and their job is to bind to DNA and turn gene expression on or off. So they are either making for more mRNA from a gene or less mRNA, which would lead to then more or less protein. So they're trying to turn those readouts on and off. They are the yay-nay for, as we just talked about, that was a good question in that context, for likely dependence on the hormone estrogen. Most breast cancer cells, if they express the estrogen receptor, that is the protein inside the cell that responds to the hormone estrogen. And they're using estrogen as essentially a signal to grow and just grow nonstop. So the idea is, if we can stop that, we can stop the tumor from growing, because it is using estrogen in a way it's not supposed to just say growth. This is what defines the use of anti-estrogen therapies. As I alluded to, PR is not the target, but it's a marker for whether or not we think it's going to be useful broadly. There's a lot of talk of precision medicine, personalized medicine. This marker in breast cancer that we've been using since the 70s and 80s is still the first, and I would argue the best biomarker we have in oncology. I think it's pretty remarkable that we've known for a long time and can still leverage that if a tumor has this protein inside of its cell, the patient is going to receive this therapy. If they don't, they're not. And it's pretty cut and dry. So we take it kind of for granted, but it is still kind of like the foundation of precision medicine. But because, and this was not an obvious thing in the 70s and 80s, if there's no receptor, if there's no ER, there's no target for the drug, so it's not going to do anything. And that still holds. All right, so that's most of what we think is going on in lobby that we're focusing there. But we still think a lot about HER2. This and the outside of the cell normally is part of what responds to different kind of cues from the environment to tell a cell to turn on and off. If a tumor is HER2 positive, it has basically copied that recipe in the recipe book dozens of times. So you're more likely to grab it, say, if you're just picking a random recipe. So there's just a boatload more message and the cell is just covered in HER2 protein. If we stain it by IHC, it's just like a dark, thick stain on the outside of all the cancer cells. Because there's more of it, it turns on all of this stuff inside the cell that it's not supposed to, and this changes what the cell is doing at the end. So whether or not is there is yay, nay for use of whether the tumor depends on HER2 signaling and whether or not the tumor is going to respond to anti-HER2 therapies. Again, same idea, no receptor, no target for the drug, they're not used. There's a lot of noise right now about HER2 low. I can't even touch that. Like, it's a weird thing, the tests aren't designed for it, but the bread and butter in the field is still, if the receptor is amplified, which means there's more copies, it's on the outside of the cell, that patient is going to benefit from anti-HER2 therapy. And so that kind of fits into the idea I wanted to bring up of triple negative breast cancer. It's called that because they're ER, PR, and HER2 negative. If they don't express ER or HER2, we don't have anything to target with our standard of care, like precision medicine. And that's why kind of triple negative breast cancer has to go right to chemotherapy and other things in that space, because they don't express the things that we would hit with precision medicine. T67 is, long story short, a summation of all of this crap going on inside the cell. All of these extracellular signals, including HER2, are turning on all of this signaling with the end goal of making that cell grow and make it divide. T67, all it does is it marks, we don't even know what it does. It's there in a cell if the cell is actively going through the process of dividing. So in a snapshot of a tumor upon diagnosis, if more of the cells have Ki67, it just means that more of the cells are actively in the process of dividing and not kind of just sitting and waiting. So yeah, this is the one that's not actually target, but it is the end result of all of this stuff inside the cell. I'll talk more about how we're thinking about targeting that, but that's what the readout is. The extra piece for Lobular that's not part of diagnosis, but we know that it's important, is e-cadherin. It's different than these other things outside of the cell. It doesn't necessarily respond to these signals, cues outside the cell, but it's really important in cells talking to each other. And I'll show you some pictures from the lab in a little bit about how it actually impacts how these cells talk to each other and how they grow. But it is connected to all of this. So we do think that, and we're trying to learn more about how Lobular is different, because if you break one of these things that's controlling all of this stuff, what happens to the cell is like kind of the short version of this. And we're only scratching the surface now of trying to understand how loss of that thing outside changes everything inside the cell.
So yeah. Is there a way at all to test for the presence or absence of ECAD?
Oh, totally. So academic medical centers will often use ECAD as a companion diagnostic for Lobular, because if a tumor looks lobular, but it's kind of on the fence, some places will order ECAD and say if it's negative, it's lobular, if it's positive, it's ductal. The problem, if that's the current definition, really, we were talking about this, I think, earlier, that the impetus for community centers to do that is not strong because a differential diagnosis of ductal versus lobular doesn't do enough yet. So a lot of centers do it. There's actually another stain that can go with ECAD to really flesh out the biology. There's another protein that moves very specifically as a result. So we can stain for lobular in that way, but how it's used is still kind of up in the air. That's a big discussion that comes up at the conference, is like, is that what's required to call lobular, or is it purely what it looks like? So it's still in the air. Okay. As I was alluding to, those biomarkers are then central to therapy decisions, and that's why getting those marks on your pathology report is so critical. Obviously, as I'm alluding to, if the tumor has estrogen receptor, and the number of percent cells positive may or may not matter, really it's like more than 1% is positive. And there's lots of, I could go on again for a while about why that's relevant and why it is relevant for biology, but if a cell expresses the estrogen receptor, we think it depends on estrogen to keep that signal to grow on. So that's why we use anti-estrogens. The currently approved therapies are tamoxifen, fulvestrant, elacestrant was just approved in January, and aromatase inhibitors. These three will directly attach to the estrogen receptor and kick off estrogen. And as a result, ER doesn't do all the same things anymore, and it stops it from changing the programming inside the cell. The aromatase inhibitors stop estrogen production, so it takes away that signal that the receptor wants to stay on. So the effective goal for all of those is to turn ER off. We don't, as I mentioned, PR is more of is ER doing this thing in the genome right now? So is a marker, but there are groups that are actively targeting, I think considering targeting PR for other reasons related to ER function. I could go on to that, but it's not prime time yet. HER2, again, we have anti-HER2 therapies that directly target that protein to turn off what it's doing inside the cell. That's trastuzumab, and now all of the other drugs, lapatinib, pertuzumab, neratinib, all of the antibody drug conjugates are almost all targeting HER2. But the idea there is, again, it's on the outside of the cell, it's doing things to the cell, so we can stop it from doing that. We can't target Ki67 because it's not really a thing to do. It's, as far as we know, it's just mechanistically, we have a very bad handle on what it's actually doing, but it's really just, is the cell growing? But what's on this is, so this is the same diagram, just with some drugs laid on. The end stage of a lot of that signaling is this protein called CDK4 and 6. And it is essentially kind of the end point of a lot of this crap going on inside the cell. And if it's turned on, the cell is going to grow. So that's why CDK4/6 inhibitors like palbociclib, abemaciclib, and ribociclib, I don't know the trade names, I forget, sorry. Those are now being used more heavily in breast cancer because they kind of are thought to circumvent guessing what's going on here and just getting to the end product. That's a simplified version, but that's the idea. With ECAD, the protein is lost. Like, as opposed to those other ones that are gained or active, ECAD's lost. So we don't have anything to target. What our lab and other people are trying to do is figure out what happens to the cell as a result and target that. It just takes a little longer to get there because it's not obvious what we're trying to hit. So stay tuned.
Matt, can you turn that on?
Oh, ECAD. So that's a good question. Got asked earlier too. Typically, ECAD hearing is deleted or lost. So the page is ripped out of the book. It's gone. Or it's, there's enough errors in it that it's not coming back. There are, what differentiates lobular from some other types of breast cancer. When breast cancer progresses, it can transiently turn off ECAD hearing, so it can do nasty things. And then it might come back on for other reasons. And then in that setting, it might be relevant too. But ductal is unique because it loses it. So turning it, like, we can do it in the lab, but it'll never be tractable. And in doing it in the lab, we don't even think it does enough because it's only one hit of many. Okay, all that I just yammered about essentially kind of like underpins the current treatment paradigm for breast cancer. If you have things change with metastatic, and I'll show you where the current stand is for metastatic, but for early breast cancer, meaning not metastatic, the question is, do you have ER? Do you have PR? Are you triple negative, HER2 positive? And all of those dictate the downstream decisions. Where chemotherapy rolls in basically has to do with decisions of risk for luminal A versus B, which very squishy is based on Ki67, but then every other risk marker that we know about, stage, grade, lymph node positivity, blah, 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 blah. There are guidelines for all of this, but it's still pretty squishy. And then the other tumor types get some form of chemotherapy radiation one way or another. And now CDK4/6 inhibitors are moving into the space of early breast cancer. I don't have a good handle yet myself on who's getting them frontline and what. Who's getting them, and there are questions of risk benefit, both from a side effect standpoint and a financial standpoint, because they are ridiculously expensive drugs. But that's another matter. Again, you'll all have these slides to review too, and the papers they come from. Okay, this is kind of the overall paradigm for like diagnosis, staging, risk assessment from early breast cancer through to metastasis and recurrence. So all of those early markers, ER, PR, HER2 are happening during biopsying. We're getting subtype information, histology for ductal versus lobular, stage, size, grade, nodal status, etc. Surgery is pretty much always first. If we take the cells out, they can't spread. That's still the first and best approach. Obviously, those are going to move on to the adjuvant therapies that we talked about, which includes endocrine therapy, the anti-estrogens, whether or not HER2 gets rolled in, and how chemotherapy and other things get involved. So we are here in this process as far as what I've just covered. What I'm going to spend my last handful of slides talking about is emerging paradigms in ILC for thinking beyond primary disease, and then where kind of biology is going, kind of in preparation for what's going to get talked about at the conference. I guess we hadn't, this is where I was going to have a break, but we guess we already had one because thank you, Larry. But any questions on early breast cancer, those biomarkers, how they relate to biology? The questions have been great. This makes my life easier trying to cover everything too. Okay. All right. Several you've already alluded to, like getting diagnosed and then getting recurrence later. This is an increasingly recognized as a unique issue with lobular, that breast cancer is already unique in that, you know, the idea of 5-year survival is such a different paradigm for other cancers versus ER-positive breast cancer. We know already overall that ER-positive breast cancer, the way it recurs just is longer, which is why extended endocrine therapy is a thing. But for lobular, it seems to be even different. And what I want to show you, so there's a lot of kind of hand-wringing about endocrine response in lobular cancer, whether or not they're anti-estrogen resistant. First and foremost, they do work. We don't have a lot of data on patients with lobular that didn't get endocrine therapy, in part because like patients weren't differentially diagnosed when we didn't have endocrine therapy. Thank you, Laurie.
Yes, thanks, sorry, I'm
So I'll talk more about Kaplan-Meiers in a second too, how these plots work, because you're going to see a lot of these. But the y-axis as it goes down is fewer and fewer patients that have not recurred. So as patients recur, the lines go down. And then this is time. So what I want you to see in the red line versus the gray line is patients with ductal cancer that did not versus did get endocrine therapy. So fewer patients recurred that had ductal cancer if they got endocrine therapy. For lobular cancer, the lines are even further apart. If a patient with lobular cancer that's ER positive, did not get endocrine therapy, their rate of recurrence is very, very rapid. Endocrine therapy dramatically benefits that. So in all the hand-wringing of does endocrine therapy work, it does. All the data we have says that it particularly works, but then it gets squishy after that. So we're seeing this now in a lot of more epidemiological data, especially since we're getting more and more studies that differentially identify lobular patients. And something is happening in the 5 to 8 year window. And I wouldn't be surprised if this is related to when endocrine therapy typically comes off or other biology. But even though these patients all had the same like risk biomarkers in these trials, something is happening with lobular rates of recurrence ticking up in this 5 to 8 year window. And what this means long-term is that the risk of recurrence for lobular patients is basically higher than all other breast cancers once you get past that 5 year window. And like things with extended endocrine therapy, we don't have a lot of lobular specific data on, so I can't like even guess is the mechanistic difference. But probably. That's why they did the trials in the first place for breast cancer overall. But most of those trials just didn't identify lobular ahead of time. So like some of the groups are going back to ask. But one of the problems that we've had in studying lobular is that when people did the trials, they just said breast cancer. And then so there is not enough statistical power to make conclusions from lobular because only 10% of the 1,000 patients is lobular. And that sounds like a lot, but statistically we can't say anything.
Is this the drug?
Oh, aromatase inhibitors is one of them. Yeah. So the field is kind of asking, like, do ILC cells, are they unique in the way they respond to endocrine therapy? Can they kind of just go dormant and wait it out? We're trying to learn that. And I don't mean to scare you, but that's what we're thinking of as a field, is trying to like understand that process and why lobular cells respond to the anti-hormones differently. But again, they work real good. No, I would be stunned if that data exists. Again, the big problem is that all the older studies, especially the no endocrine therapy trials, are before anybody bothered diagnosing. So it's a beast to even ask those. So there's like different therapy decisions for pre versus postmenopausal, and the biggest trials that were done to date, they have been going back to identify lobular patients, but again, the issue is that they're grossly underpowered. So even if they come to a conclusion, they're not gonna make a big deal about it because they don't have the statistical power to be confident. It's a huge problem.
So how do we fix them?
Meta-analysis, yeah. That's
very complicated examining the question is 5 years, is 8 years better than 5 years? So the trials aren't happening, and more trials that are happening now do have lobular subgroups specified. So it's happening slowly, not as quickly as any of us would like it necessarily, but
I really find out about trials, so, because it's hard to differentiate. I mean, you go in the oncologist's office and you've got, mine does anyway, has, different trials posted on the wall. But how do you know, without looking at every single one, what would apply to you and what you should call on? Because there are disqualifiers of some of those clinical trials, and haven't seen one yet that's ILC specific.
So we should come back to that. So LBCA does have a trials listing for lobular. It's the bigger, I mean, it's hard. So many have exclusion criteria. A lot of big trials actually do exclude lobular
Yeah.
specifically. So it's a thing that we could talk about separately, but yeah. We've already seen a bunch of these type of plots, these survival curves. I just wanted to spend a minute because like these are obviously the things that impact you all the most, like end stage. So let's talk about what they're actually representing. So they can go up or down, and one way or another, it's either proportion of survival, and I'll come back to that, or proportion with an event. So if it's going down, it's showing fraction without an event. If it's going up, it's showing a fraction with an event. And it's just purely a stylistic thing that the investigators are showing. And then time is always on the x-axis. Maybe it's important that it's to look at, it could be weeks, it could be months, it could be years, depending on the type of cancer and the intervention they're looking at. Survival is very hand wavy and is very study specific. So this basically means the patient is alive and hasn't experienced an event. We had event-free survival earlier. Event is a very specific thing for a given study, and I don't know if that poster actually decided. So there are this is kind of 4 typical types of events that are considered in these plots. So progression-free survival. In that case, the event means progression of the cancer, like lack of response to therapy, growing in size, spreading to another site. Those are all markers of progression. And a study has to define what progression means. And it's fairly uniform, but can still be study specific. Distant metastasis-free survival, the event specifically means that a metastasis was diagnosed away from the primary site. A local recurrence in the breast doesn't count as an event there. Breast cancer-specific survival, the event is death due to breast cancer, which is usually defined by the study how they determine that. And then overall survival, the event equals death due to any cause, which may or may not be cancer-related. So all of those things lead to different curves, and patients then may have an event by one definition and not in another. But then what happens with these is that where the line is horizontally relative to the y-axis is the fraction of patients alive and still on study, because I'll show this in a second. Patients can either like fall off the analysis because of a different event, or they're lost to contact, which is another thing. So where it's at is fraction of patients that are alive and still on study without an event, and where it is on the y-axis is recalculated every time there's an event. So patients can be lost to follow-up in the interim, but it doesn't affect where the line is until another event is registered. And that'll make more sense, I think, in a second. So this is a Kaplan-Meier plot from the late 2022 paper that led to elacestrant getting approved as the latest endocrine therapy in our arsenal, in this case for ER mutant breast cancers. So their event in this analysis was progression-free survival. So if a patient had progressed on therapy, again, meaning increase in size, they probably had some imaging protocol, or distant metastasis, and other definitions, those are all progression. So that would have counted as an event. So you start at 100. In this case, they have number at risk. They had 115 versus 113 patients in their experimental arm versus the standard of care, which is what SOC is in this case. And then every time there's an event or a patient experiences, in this case, progression, the percent of patients with progression-free survival goes down. So the confounding thing there is that in this case, circles, sometimes it'll be hashes, are censored events. And these are when a patient is either lost to follow-up or has an event for another reason that doesn't qualify for this. So if a patient died of a heart attack that was unrelated to breast cancer, they'd be censored in this analysis, because you can't say that they would have had progression-free survival longer, but they didn't have an event as qualified in the study. So that's what the circles are. And those are more often than not because they lost track of the patient, is what typically it is. So down here then, the number at risk is the number of patients they started with, minus any events, minus any censoring that happened. So that kind of changes the denominator every time there's an event then, which is why the lines can get screwy as you go farther out. Because in this case, there might have been, like at this drop here, there might have been only one event, but then they had a bunch of censored, so it changes the math on who's left. Don't worry too much about it. Point being, every time it goes down, something happened in terms of progression or survival. What they're looking for then is a separation in the curves between an experimental approach and the standard of care. Typically, we're talking about median survival, which is when 50% of the patients have not experienced an event or just at that point. So in this case, they're comparing 50% of patients having progressed on standard of care versus 50% with the experimental therapy. And they're doing statistics to say, is this rate different than this rate? One thing you can see here, though, is that there were a whole bunch of events early. This probably means that those tumors were never going to respond to this therapy in the first place. So once you get past this point, then you have the tumors that are going to eventually develop resistance. And there's a lot of reading between the lines in these plots, because this is stuff in the clinic, not stuff we can track in the lab necessarily. So there's a lot of stuff in the middle. This is from the same study. I like stretched them so the time frames match too. It's the same exact study, same exact treatment. On this one, the event, it's progression-free survival. On this one, it's overall survival. So these curves drop much more quickly than these, right? The reason being, like, so they go down early, the curve goes down. But after progression, patients are going to go on to other therapies. They're going to get other chemotherapies. They're going to go on other clinical trials. So they won't have been they won't have necessarily passed at that point of progression. But that's why this one looks like it falls off much more quickly than overall survival, because this is related to, like, they're basically off study at this point. They're just being tracked for outcomes. So, but that's why the axes are very different and why the shapes are very different. Back to biology. I promise I don't have a ton more. I showed the current paradigm for early breast cancer. I wanted to show where things are at for metastatic disease. This was just updated in March with essentially 2 new therapies in the metastatic space. So this is really hard to read. You will get the slides. And this is also on ASCO's website. Basically, there's decisions on, first off, whether or not there's a germline BRCA1 or BRCA2 mutation, because we have treatments that specifically target that biology. And stuff that my lab is actually studying. But so that the talazoparib poster we talked about earlier is kind of applying these ideas to lobular cancer. And I could go on about that for like another day. Aside from that, though, for ER-positive metastatic disease, did the patient get an aromatase inhibitor? Yay or nay changes the choice for the second line endocrine therapy, which is either going to be, if they didn't receive one, the patient will get an aromatase inhibitor. If they did, it'll go to fulvestrant, which is another anti-estrogen. Patients are typically getting CDK4/6 inhibitors now as frontline for advanced or metastatic disease. And then there's decisions about whether the tumor has hits or mutations in a gene called PIK3CA or estrogen receptor. And there are targeted therapies for both of those. And then after those fail, then there's a whole slew of different chemotherapies that get brought to bear.
So I experienced with genetic testing, those are they test for.
Those are routine now. Like the ER mutation one is routine as of like January. PIK3CA isn't much longer, but those are part of standard panels anyway.
Are they part of initial diagnosis or recurrent gene, right? So you're looking at PIK3 on your oncotype score, it's only if you have a recurrence.
Yep.
So, and I'll just add to, like, if you have progression, they can retest, or especially they can get cell line. Because these are one that you actually can develop.
But if you do a separate genetic test, like if you're really big BRCA1,
Oh.
they're not testing for the.
So yeah, BRCA1 is it's called germline, so they'll take a blood sample for that one. It's totally different, but good question.
So what was the update? March 23.
This one. Yeah, that was that elacestrant drug I just showed you. That was literally just happened. Okay, the last like handful of slides are just general themes. Like this is like words you're going to hear at the ILC conference and in the science moving forward. So when we talk about cancer, the simplistic view is that it's a lump of cancer cells. The reality is that it's much more complex than that. There's a whole what's called the microenvironment of the tumor. This is kind of the textbook view as of like 10 or 15 years ago. The gray are cancer cells. You have immune cells that are in there as part of the body's either functional or dysfunctional immune response. You have blood vessels, you have lymph vessels, you have structural support normal cells that have been like hijacked, and they're called fibroblasts or cancer-associated fibroblasts. And then the microenvironment's different between the primary tumor and the breast, say, versus a metastatic tumor somewhere else. So you hear a lot about tumor microenvironment and tumor immune microenvironment. This is 10 or 15 years ago. This is where we are today. And kind of the big changes are like things like cancer-associated fibroblasts are kind of normal cells that, because of them being adjacent to the tumor, now behave differently. And there's now like, I don't know, 4 or 5 different categories of those cells that we're talking about as far as what they do to the tumor, how they talk back and forth. And then the immune landscape, this is over my head. But there was a whole slew of the big classes are macrophages and T cells, which are just different immune cells that do or don't get recruited into a tumor. The tumor can either shut them off, or if you know, in the best case scenario, they're on and fighting the tumor. But then there are spectrums of what that activity means. And we are learning that for all of these things, that lobular is different, which I'm sure you'll all be very surprised to hear. They do talk to those fibroblasts differently. There's at least 1 or 2 talks on that at the meeting. And then the immune microenvironment, the cells that are actually in the tumor and lobular seem to be different than other breast cancers. And we're only just starting to understand what the implications for that are on biology and such. Tumor heterogeneity is also a thing. And so up until now, I've been telling you this story of like cells progressively acquire hits and become cancer, and it's a progressive process. The reality of it is that any one group of cells that acquires hits, like this group over here can acquire another hit and go this way. This group over here can acquire a different hit. And really in the end, a tumor is a grab bag mix of hits, and they can represent different clones that are called, essentially cells that are genetically different, but all part of the same tumor or mass. There's been some really cool studies in lobular lately to kind of flesh out how things may or may not be differently than ductal cancer. I'm not going to go into depth in this, but suffice to say, what we can do is look at the different hits that are in primary tumors versus metastatic disease, count the number of hits, which hits are there, when they appear, and kind of develop an evolutionary tree for when the primary versus metastatic, when they left each other, when they spread. And long story short, we're learning a lot about different mutations, when they happen, how that like affects the relative composition of a tumor, and how metastatic disease is different from the primary tumor. It all falls under the umbrella of tumor heterogeneity, which is a big topic in the field. Obviously, metastatic ILC is also different than other breast cancers because in part it does go to some of the usual sites, but it does go to some different sites. That's another big topic at the meeting. Lobular, as we've alluded to a bunch today, can go to a bunch of sites in the abdomen, and we think it's part of why how it grows differently is that it can survive in that space. So this is just some data on how we think it's happening and how we're learning about it. All right, just a couple more slides. We've talked about e-cadherin a lot. We know it's central to ILC genetics, but we don't know why or what it's doing to the cells. So this is, Donna, you asked about the staining. Can we find e-cadherin? This is e-cadherin staining in a ductal cancer. You can see, I mentioned to you that e-cadherin lives on the outside of cells, and you can see that in the stain. You can see these little blue dots are actually the DNA, and then there's a brown ring kind of somewhat around that. So that's the border of the cell with the DNA in the middle. And that's on all the ductal cells. You can see that brown stain, which is where e-cadherin is. On the lobular cancer, this is actually a normal duct next door to it. These are the lobular cancer cells, and they're dead negative. They have no staining whatsoever. So they don't have any of that protein on the surface at all. And that is kind of, if we have to differentially diagnose ILC, that's how it's done. This does translate to what the cells actually are doing in the lab even. So if we take a ductal cancer cell line, those cells that grow in the Petri dishes, in this case, we're forcing them to grow so they can't grab onto the dish, the plastic surface they're on, so they kind of have to talk to themselves. The ductal cells are kind of fighting to survive. They don't like not being able to attach to things, and they form these little spheroids, they're called. They're like little lumps. They look like little tumors. But the ductal cells don't like being in those conditions. They want to be able to grab onto each other and to a surface, and they form these lumps. Lobular cancer cells don't do that. They don't care. They don't tack to each other the same way. They do not form lumps or spheroids. They instead are these like loose, disaggregate, discohesive messes that just kind of float in the dish. And if you like squint and project it onto histology, that's what they're doing in the breast too. They're just kind of snaking through, and they're not really talking to the surrounding microenvironment. So we do think that this is not just a biomarker, that this is a key part of what makes lobular tumors different than other breast cancers. And I alluded to this earlier, ECAD is connected to all of this stuff that I talked about. But the field is now moving beyond biomarker and saying, what is the biology? How does ECAD make lobular happen? Yeah. So this was supposed to be before lunch. We're after lunch now. But I literally just covered like a semester of molecular and cell biology and breast cancer. So any questions? Yeah.